And welcome again to the evening session of the L. Russ Bush Center for Faith and Culture's Conference on Evolution, Genetics, and the Historical Adam. This event is sponsored by the L. Russ Bush Center for Faith and Culture, and I mention that again because Dr. Bush's widow, Cindy Bush, is with us. She has joined us this evening, and so, Miss Cindy, we are honored and glad to have you with us this evening. We have two very able speakers who have done a spectacular job of communicating some very difficult things to us. For those of us who are non-specialists, which that's all of us in this room except for those two men, I suspect they have done a remarkable job of presenting some very complicated things in ways that we can get, and I do appreciate the way that you have presented yourselves. Now let me just mention one more time, Dr. Jeanson's book in which he contributed is Searching for Adam, Genesis, and the Truth About Man's Origin, edited by Terry Mortensen. Copies of the book are outside the door, along with some other books. Dr. Jeanson may mention that here in just a little while. Dr. Venema's book, he has flown down from Canada, and bringing books is not quite as easy for him to do, but it is available at Amazon. It is Adam and the Genome, Reading Scripture After Genetic Science. Let me encourage you to purchase both of these books, and that way you'll be able to read their arguments and get what they're saying. If some of it you kind of say, okay, I need more of that, they elaborate on these, on the arguments they're making here. They're in these books. So we heard first from Dr. Jeanson and then from Dr. Venema in the afternoon session. Now in this evening we're going to switch the order. We're going to hear from Dr. Venema first and then Dr. Jeanson. Between the two men we will have a brief break, and then after the two of them have spoken we will again have just a brief break, and we will set up the front in order to have a dialogue in which we will have both men interact with one another and with the questions that you have sent. And so this is being live streamed. We're excited. We've had almost 10,000 different people watch the presentation this afternoon, which is very exciting. So if you are, let me encourage you, if you're wanting to submit a question, and I encourage you to do so, go to Southeastern's Facebook page. You'll see it being live streamed. In the comments section, write your question. We will try to get to as many questions as we can during the dialogue portion. But that will be the way that questions will be submitted for this event. Dr. Venema, we are glad to have you with us. Would you join me in making him welcome one more time?
Good, appreciate it. Okay, good. It's a delight to be back, and hope you enjoyed your dinner, as did I. Is there something about Chick-fil-A that just is part of the Southern experience, or? We don't have Chick-fil-A where I come from, so hopefully the Vittel Agenda discussion didn't create any ethical concerns for you as you enjoyed your chicken sandwich. Okay. In the last talk, I discussed the heliocentrism issue. So what I'd like to do is actually return to that a little bit as a way to think about how Christians in the past have negotiated some of these sorts of issues, similar to what we are facing now. I sometimes tell my students, lucky you get to live at a time when the church gets to wrestle with these sorts of serious questions. We haven't done this for a couple of hundred years. And they say, I don't want to live in a time when the church wrestles with these kinds of questions. I want to live in a time when the answers are there for me. But such as it is, God has appointed that we live in this time and in this place, and this is the time that we are given. And it is a time when the church is wrestling through these things. Just as an aside, though, I think that as long as the church can learn to dialogue graciously on these points to the best of our ability, and we're not perfect, we're humans, right? So we are going to make mistakes, absolutely. but to the best of our ability, if we can dialogue graciously on these issues, at the end of the day, whether one accepts one version or the other, if we recognize one another as brothers and sisters in Christ, and then if the world can see that we can dialogue charitably on these things, then it won't become a missional problem. It won't be something that people on the outside look in and say, oh, look at those Christians fighting over these things. We're meant to be known for our love, and this is an opportunity where we can show love. Now, I understand that, for the different denominations and whatnot, can be quite a struggle in certain areas. I don't diminish that. But if we can do it in a charitable way, I think if we can learn from history on that point, we'll be ahead of the curve on this one. Okay, let's move back to something that is not theologically challenging for most of us in the present day. So if we look back in history, we can see that the idea that our solar system, and even to use the word solar system is to imply sort of a Copernican way of looking at our universe. Nicholas Copernicus was the first individual to propose sort of a coherent mathematical model of a heliocentric solar system, and you can see when he lived. So up until that time, the view of how the universe was constructed was not viewed to be heliocentric. The notion was that the Earth was at the center of the universe. It was a geocentric or Earth-centered system, and everything went around the Earth. So Copernicus's idea that the sun might be the center was viewed as something that was a new sort of mathematical way of thinking about things. And it was also, it kind of came onto the radar of the church as well, that perhaps there was, something theological here that might be concerning. So this is a very famous quote from Luther. Now, I don't want to press Luther too far on this particular point, because Luther wasn't a scientist, and as far as we know, this was kind of just like an off-the-side, kind of off-the-cuff kind of comment. So this isn't perhaps as he's had opportunity to reflect deeply on this, but he says there's talk of a new astrologer. So astrology back then, that's actually not a bad way to... astrologer now doesn't mean what it meant back then. So there's a new astronomer who wants to prove that the Earth moves and goes around instead of the sky, the sun, and the moon, just as if somebody were moving on a carriage or ship might hold that he was sitting still and at rest while the earth and the trees walked and moved. But that is how things are nowadays. When a man wishes to be clever, he must invent something special, and the way he does it needs to be the best. The fool wants to turn the whole art of astronomy upside down. However, as Holy Scripture tells us, so did Joshua bid the sun to stand still, and not the earth. Copernicus didn't really encounter too much resistance. And again, there's a lot of history here that makes for very fascinating reading, but one of the things that we can see in the response to Galileo, who was the next individual to come along and sort of further this line of investigation in a major way, we can see in the responses to Galileo that Copernicus's views weren't seen as particularly theologically threatening because it was thought that he was just sort of proposing a hypothetical model, kind of a mathematical model to describe how, a solar system might work mathematically. But it wasn't viewed as particularly threatening because it wasn't seen to have much traction in reality. So it's a nice model for the philosophers and the mathematicians, but it doesn't really work that way. Well, Galileo comes along in the late 1600s, and it's actually remarkable to think that we're only about 400 years removed from that time. It just shows you how quickly science advances since that time. And Galileo actually had some observational evidence that he could bring to the table. So he was one of the first individuals to construct and use a reasonably high-powered telescope. And with that, he observed the moons of Jupiter, and he also observed that Venus has phases. Actually, I was on a hunting trip many years ago in the north of British Columbia, and it was a nice winter's night, and I was sitting around the campfire with a bunch of guys after a day's hunting. And if you're a hunter, you might know that, sometimes people have spotting scopes, which are basically low-power telescopes. And I happened to notice that Jupiter was visible, and I said, we should set up Gary's spotting scope and have a look at Jupiter, because I bet you we can see the moons. So we did, and we had this beautiful experience of looking at the moons of Jupiter, and then we had this fantastic conversation about science and faith. And not everybody in the hunting party was, of the same mind as me, and I became painfully aware that there were many firearms around the location at the time. No, it all worked out fine. It was an example to have charitable dialogue, and also to appreciate God's creation. But what Galileo saw as he looked at Jupiter was that it has small moons that go around it. So here we have, and we still call them the Galilean moons to this day, the 4 that are visible in that manner. And what that did was it sort of provided in microcosm what a solar system might look like. Here we have a body, at a center, and we have things going around that. So could that potentially be a sun, like a model of what the sun and the earth look like? The other thing that Galileo observed, one other thing that I don't have on the slide here, is that he saw that the moon had mountains on it, that the moon was not a uniform sphere. And up until that point, everybody thought that everything celestial was perfect in the sense that it was a perfect sphere. So that was theologically troubling as well. I know it seems a little bit odd to think about these points of theology that don't have any traction with us, but they had quite a bit of traction back then. The other thing he observed was that Venus has phases, just like our moon does. So Venus waxes and wanes. Venus will appear at sometimes as a full Venus and as a crescent Venus. Now, that is something that cannot be squared with the previous model of geocentrism that was prevalent at that time in that place. So this observation pretty much ruled out Ptolemaic geocentrism, but an additional model was also developed at this time by Tycho Brahe. And he had a different model. So this is a model wherein the Earth is still the unmoving center of the system, but the sun and a few of the planets, the sun is the center of a few of the planets, but then the Earth is still the unmoving center of the entire system. Brahe's model is basically the idea that if you could reach in from the outside and grab the Earth and hold it still and let everything else go whirling around it, that's the mathematical model that Brahe was proposing. And there's actually no way to distinguish between those two models, or there wasn't at the time technologically. But it was theologically a more satisfying model because of the theological commitment to an immobile Earth. So what we needed was a different point of view, and we needed a different point of view for the Earth itself. And this has to do with something called stellar parallax, this idea that if the Earth actually was moving through space, we should be able to see this particular effect when we look at stars that are close to us relative to stars that are far away. So I sometimes get my classes to do this, so I'm going to get you guys to do this too. If you want to observe parallax, take your hands, put your fingers up with me. If everybody does this, then I won't feel as foolish. There you go, although I'm the only one on the live stream, right? Okay, so put one close to your face, and then put another one in line farther away. Now close one eye, and then wiggle your head back and forth. You can see that the relative position of your two fingers seem to switch places with one another. So what was expected was if the Earth is going around the sun, that our perspective would shift. So a nearby star and a faraway star, as the Earth is going around the sun, we should be able to see the relative position of stars shifting to one another. And that would be very good evidence that the Earth was in fact in motion and not the sun. So the idea was plain, and it was easy to see how it could be tested, but technologically there was no way to do this back in the 1600s, just because you need very high magnification, powerful telescopes in order to do this. There wasn't an appreciation for just how far away the stars are. Can you imagine trying to detect parallax if I held two fingers together like this, and then I stood across the street and you wiggled your head back and forth a bit? You wouldn't be able to see it. Now, you might be able to see it if you had a very high power telescope and very precise ways of referencing, but it wasn't accessible to them at this time. Now, the inability so we were able to observe this in the 1800s, and there were actually other convincing lines of evidence that came in prior to that time as well. But the inability of scientists to observe this stellar parallax in the 1600s was taken as confirmation that the Copernican opinion, as it was called, was not only unscriptural, but also poor science. So there was this expectation this observation should be made, and it was felt that that the tests had been done and the observations had failed. So let's go back to John Edwards from the 1600s. I quoted him on solar fusion. Well, he didn't know he was talking about solar fusion, but we quoted him previously. So here's John Edwards, 1696, discussing theologically what the issues might be with respect to heliocentrism versus geocentrism. So he says, the Copernican opinion seems to confront a higher principle than that of reason. So languages have shifted a bit, so it's going to take a little bit of translation as we go through, but of course language is shifting was something that we talked about previously as an example. But he's basically saying there's a higher principle to that of science or reason. And when the chips are down, you go with the Bible. You don't go with science. That's basically what he's saying. He says, if we will speak like men of religions and as such own the Bible, so own, like do you own it, as in do you stand on its authority? We must acknowledge that their assertion is against the plain history of the Holy Book. For there we read that the sun stood still in Joshua's time and went back in King Hezekiah's. Now, and this is the same passage that Luther was referencing as well. Now, this relation is either true or false. If it be the latter, then inspired Scripture is false. So what Edwards is willing to say is, if you can prove to me that heliocentrism is true, then that indicates to me that Scripture is false. It says, if it be the former, i.e., if the relation really is true, then the sun hath a diurnal motion about the earth. For the sun standing still could not be strange and wonderful unless its general course was to move. So for Edwards, if you have to make a choice between science and Scripture, between reason and the Bible, then you go with Scripture, not with reason. Okay, now, that's obviously not the situation in the present day. Most of you are not losing sleep over the heliocentric nature of our solar system. But have you ever paused to wonder why you are a Copernican Christian? Or why, for me growing up, Darwin was a bad word at church. If you heard the word Darwin, that was bad. Evolution was a bad word. You don't talk about that. But Copernicus, no big deal. Copernican never read, if somebody called somebody a Copernican, not that anybody ever did, it wouldn't have registered as a bad thing. But have you ever wondered why that's the case? Interestingly, there is no scriptural support for heliocentrism. And if there was, we would have seen those texts brought to bear in the conversation that Galileo and others engaged. There simply is no scriptural support for heliocentrism at all. It's just not there. At best, you can argue that Scripture is silent on the issue. Now, I'm not 100%, I looked at some of Answers in Genesis' materials on this case, and I believe, I may be wrong, maybe Nathaniel can correct me, but I believe that this is the position that Answers in Genesis takes, that Scripture is simply silent on this issue. Although that is very much not what was thought to be the case back in the 1600s. They very much thought that the verses, one example there on the bottom from Ecclesiastes 1:5, they very much thought that these verses had a bearing on the discussion. So, how is it that we've made our peace with Copernicus? Perhaps you don't even know. Perhaps you didn't even realize that there might have been this issue to make peace with. Although, interestingly, there are still a very few Christians who do hold to a geocentrist view, and they can be found. So, for that matter, we've got NBA players that hold to a flat earth, right? So, okay. So why is it that we're not troubled by Copernicus? In the present day, most Christians are perfectly comfortable with the idea that Scripture in some ways speaks to the original audience in a way that the original audience would have understood it, such that it would not have been a barrier to their understanding. So one way to think about this is that Scripture is speaking in a phenomenological manner. So we even say this now, the sun rises, the sun sets, as it were. Even though those things are scientifically false, as it were, the sun does not rise, the earth turns, the sun does not set, the earth continues to turn, and so on. But that's phenomenological language. It's from the perspective of what we observe, and it's not considered untruthful to speak about things in that way, right? Nobody says, Dennis, you're a liar when you say that it's a beautiful sunset. Okay, so, and of course the analogy there to evolution is fairly straightforward. We should at least as Christians be open to the idea that perhaps Scripture is speaking in a phenomenological manner and speaking in a way that the original audience would understand. So when we hear in Genesis that God is stating that different species are being created or different organisms are being created according to their kind, and kind being defined as reproduction, it's not surprising that that would be a phenomenological way of describing things. The ancients would not have had an access or an understanding to larger spans of time to understand that perhaps species might shift over large periods of time. Okay, so the 1800s saw significant developments in biology, namely Darwin's theory of evolution by natural selection. Darwin was the first to provide good evidence that modern species are derived from ancestral species through a series of transitional forms, and we've seen in the previous lecture how that particular hypothesis continues to garner very strong support from modern scientific enterprise. Like heliocentrism, evolution has encountered resistance from within the Christian community. Evolution is fairly widely doubted among Christians. It doesn't take much effort to look at surveys of North Americans, find out what their opinions are on evolution. And this is a theory that is widely doubted by Christians. And most Christians feel that there's insufficient evidence to support it as a viable scientific theory, especially for human evolution, right? If you look at the different sort of Pew Forum survey results, there's usually more resistance or more reluctance to say that humans might be a part of this particular process. As we saw in the last lecture, though, modern-day genomics nicely fits with other lines of evidence that we've seen, and that evidence is quite substantial that humans do indeed share in this process of evolution, that we are the products of an evolutionary process. Now, what I'm going to speak to now is that not only did we evolve, not only did humans come about through, not only did God bring about humans through an evolutionary process, but that also took place as a population rather than starting from a single ancestral couple. Now, our observations, oh dear, qu'est-ce qui s'est passé? All right, don't send. And you get to all watch me as I try to get this. I have no idea why that just happened. I was joking with Nathaniel earlier that if we had technical problems in my presentation, that perhaps some would see it as a sign of divine displeasure. So. I'm glad you have a good sense of humor. All right, where were we? Okay, I think we were here. All right, Lord willing, this will work. Okay, so let's just briefly review. We had Darwin's ideas about him proposing that species share common ancestors, the idea that populations become genetically separated. We've got changes, so this is just review from the previous talk. We've got these different changes that perhaps can accumulate, giving a shift in average characteristics over time. The differences may lead to new species over time. And then we drew that analogy to languages, how languages shift over time, populations becoming separated, differences showing up, average characteristics changing. And we made the point that both languages and species are information systems that are copied imperfectly, so we have this opportunity for change. And then I showed you these texts to sort of illustrate this process using language as an example. Now, here's the point that I want to make here, is that languages don't start with two individuals heading off and starting their own language. Likewise, species don't start, typically, with two individuals going off and starting a new species. Or languages don't start when two individuals start suddenly speaking a different or a highly different version of the language that they spoke before. Species don't start when there's some sort of dramatic mutation that makes them different from the previous organisms that they were a part of. So evolution, like with languages, is all about average characteristics of populations shifting over time. Now, so the idea that any large mammal, us included, would have started as a population of just two solitary individuals would be extremely unlikely, even in the absence of any sort of genetic evidence. And that's because population shifting over time is how evolution works. It's a gradient. Okay. Now, I made this, I'm not sure if I showed you this slide or not, but one way that you can tell if species are connected with one another is we've sort of seen some of these lines of evidence in the previous talk. We can go around the world and easily find related languages. I use this example in the book. I grew up with one set of grandparents as Frisian speakers with a last name like Venema, or if you're Frisian, Venema. That it's not surprising that some of my ancestry comes from the Netherlands. And what was interesting to me as a child, I relate this somewhat in the book as well, is that I would notice my grandparents speaking West Frisian, and I would notice how uncannily similar some of those words were, relative to English. And what I didn't know then, but I do know now, is that modern-day English speakers and modern-day West Frisian speakers — Friesland is a province of the Netherlands with its own distinct language — those two language groups share a common ancestral population of speakers that lived at about the year 400 on the continent. And then some of them populated the British Isles and some of them stayed put. So modern-day Fries and modern-day English are about as closely related as two languages can be, but yet they are separate languages. And this sentence in English and in Frisian are pronounced almost essentially the same. Butter bread and green cheese is good English and good Fries. It's a nice little pithy saying. And the pronunciation in both languages is almost identical. But again, the point is that these populations became distinct over time, and that it wasn't just that two individuals went and started English and left the population of ancestral speakers on the continent to become Frisians. Okay, so modern genomics indicates not only that we arose through an evolutionary process, but also that we did so as a population. The evidence is such that at no time in our evolutionary history has our ancestral population size been below about 10,000 individuals. Now, this is based on looking at measures of current genetic diversity in humans. So the idea is that we look at present-day humans. We understand processes of recombination, mutation, and that sort of thing. And we can use that to infer in the past what our ancestral population sizes might have been. Now, there's a number of different ways that you can do this, and I go through a number of them in the book. All the methods that we've applied to humans so far agree that we descend from this population of about 10,000 individuals. Now, it's a very common question at that point for people to say, well, where did those 10,000 come from? It's a little bit like asking, well, where did the first English speakers come from? Well, they would have come from a population that was almost identical to them, and so on, and so on, and so on in the past. So these 10,000 individuals don't sort of just drop into existence or pop out of nowhere. We're talking about a population that if we go farther back in time, eventually we would share common ancestors with species like Neanderthals, or I should say Neanderthals now, that's the proper way to pronounce it now. And then species like the Denisovans, Homo erectus, apes in general, chimpanzees, gorillas, and so on. So the idea is that we've had this long lineage that has split along the way, and at no time point in any portion of that whole lineage has our population been below about 10,000 individuals, measuring back at least to about 18 million years ago or more. Okay, so how do we go about doing this? How do we measure population sizes? There's a number of different ways to do this, and it's decidedly tricky to try to explain this sort of material. It's one thing for me to show you a picture of vitellogen and pseudogenes in the human genome. It's like, wow, that's very qualitative evidence. I can see it and I can understand it. This evidence is quantitative. It's based on variation, a DNA variation that we see in present-day organisms. So it's a little bit harder. I can't just show you a nice picture of, like, say, a pseudogene or something along those lines. Here's one method of estimating population sizes that employs genetic markers that are linked close together on chromosomes. So if we use just a line to represent a chromosome and we put little hash marks on it to represent different locations on a chromosome, and then we'll use different letters to indicate different DNA letter variations, that are present in those different spots. So those variations could be as small as one single DNA letter difference between you and me, for example. If we look in the present day, and this is just a large population of individuals, this is a pedigree. If anybody's had a genetics class, maybe you've had to deal with some of these pedigrees. So squares just indicate males, circles indicate females, and then the lines just indicate marriages and descent. So if we look in the present day generations, the most recent ones, we might find a number of individuals that have the same genetic markers in the same spatial pattern. Now, there's a couple of different hypotheses for how that might come to be. It could be that mutation events independently mutated to provide those different patterns in the different people. That's not very likely, although it's formally possible. Another possibility is that recombination events that sort of, you might remember crossing over in biology, sort of chromosomes getting together and then breakage and rejoining to mix and match different variation between different chromosomes. It's possible that different recombination events produce that. Again, possible, but not very likely. what's more likely and much more probable is that these individuals have inherited this little snippet of DNA with these different variants from some common ancestor that they share in their past. So they all inherited it from their great-grandpa or something like that, as opposed to independently assembling it. So you can choose markers that are really closely spaced on a chromosome, and the probability of recombination, mixing, and matching those is quite low, because the closer things are, the less likely you'll have that crossover event that happens. Or you can also take marker pairs that are farther apart. And if you take those marker pairs that are farther apart, they actually will recombine more quickly, and as a result, they will mix and match more quickly as you go back. So if you take marker pairs that are really close together, it allows us to look a very far distance back in human populations. And if you take marker pairs that are farther apart, then it allows you to look at shorter distances. So one of the nice things about this particular way of doing this is it allows us to scale it relative to time and look back over the last few 100,000 years to see what population sizes may have looked like for humans as we became human. Now, these marker pairs, not that you're going to be able to see that, but what you're seeing there is a phylogeny, or a tree of relatedness, for present-day humans. So these are humans that live, in the present day. At the bottom of the graph would be various African populations. And actually, it's non-African populations that are at the very top. The bulk of this is African populations. You might not know this, but the bulk of DNA variation in modern-day humans is in sub-Saharan Africa. Anybody who's not sub-Saharan Africa comes from the out-of-Africa migration that happened at about 60,000 years ago, plus or minus. And it's a very small population that moves out of Africa and then populates the rest of the planet. So if you're painfully white like me, then that means that you've got less genetic variation than our brothers and sisters in sub-Saharan Africa. Okay, so maybe that's why white guys can't jump. I don't know. So these different patterns of genetic letters, they correlate with what we know about human relatedness. So we see different ethnic groups around the planet that have these little patterns of markers, and they have patterns of markers that are consistent with their ethnic group. So these markers are behaving as we would expect them to behave when we look at DNA sequencing around the planet. Now, this kind of thing is a computational biologist's, they're amazed by this. The ability, this is their fantasy that you can have this amount of data to work with to describe different human populations. So you won't be able to see this, but what this is looking at numbers of marker pairs for different chromosomes in the human genome, and then simply asking the question, based on the diversity of marker pairs that we see in these different populations around the planet, how many ancestors do you need to account for that particular people group? So at the top left there, you've got human chromosome 1. The first column is no, it's not a sub-Saharan African population. It's some non-sub-Saharan African population. They looked at 1.3 million pairs of these markers on the first chromosome alone. They crunched the numbers, and the estimate is that it takes about 2,800 individuals to be ancestors of that particular people group relative to that chromosome. And what you can do is you can just go around the planet and do that for everybody on the planet, all the different people groups, and then just add up the numbers. And when you do, it comes to that about 10,000 individuals for the planet as a whole, for humans as a whole. What's really nice about this particular way of doing things, maybe I'll just go back for a second, looking at different marker pairs like this, is it's a way to estimate population sizes that's independent of mutation frequency. Because these are not new mutations that are happening. This is just the segregation and recombination of already existing mutations. So it's one way to do this analysis without having to make an estimate of mutation frequency. Now, you can do estimates of population size that are based on mutation frequency, and those estimates also come to about 10,000, but they use different methodologies and have independent assumptions. Okay. So that gives us some confidence because we see these different methods of estimating population size that use different assumptions, but yet converge on that same answer. This gives us confidence that what we're looking at is, in fact, accurate. There's another method of estimating ancestral population sizes that actually neither uses mutation frequency, nor does it use estimates of recombination frequency. Now, we can measure mutation frequency in the present. We can measure recombination frequency in the present without trouble. But there are other methods available that actually don't even use those things. And this is one particular example that I like to use. One method of estimating population sizes uses the pattern of relatedness among closely related species. So in some cases, what we will see is certain gene variants that don't nicely fit with the overall species pattern. So I've shown you evidence previously that humans are most closely related to chimps, and then next most closely related to gorillas. Now, one thing to keep in mind is as populations separate and go their separate ways, as they speciate, they have genetic variation within them while they're undergoing that process. Okay? So there's going to be genetic variation present in a population as it's undergoing a speciation event. So what that means is you can have this particular effect take place. This is something called incomplete lineage sorting. I talk about it in the book, and it's a bit challenging to understand, but I'll do my best to walk you through it. If you look at the yellow variant, so here's two different DNA variants, yellow and blue, in this ancestral population before it splits to become the common ancestral population of gorillas and the human-chimp common ancestor, and then later on it also splits. The human-chimp common ancestor population diverges to become present-day humans and present-day chimpanzees. The yellow and blue DNA variants are present in the population of the common ancestral population of all three species before it goes through this process. You can see that the yellow DNA variant is retained in the human lineage, and it's also retained in the gorilla lineage. The blue variant, on the other hand, is lost in the human lineage, and it's lost in the gorilla lineage, but it's retained in the chimpanzee lineage. So what this means is for this particular little bit of DNA, the human genome will actually more closely match the gorilla genome rather than the chimpanzee genome. Okay, so this is called incomplete lineage sorting, and the idea is that not all the variation in a source population will equally sort down. Okay, that's a bit confusing, and it's genetics. Let's take an example, an analogy, and language can help us here again as well. So here I am, a Canadian, speaking to you as Americans. So I don't need to tell you that Canadian and American English do have a few differences, but if you've ever been to the United Kingdom, you would know that the differences between Canadian English and American English are rather small compared to the differences you would experience if you went to the United Kingdom. So Canadian and American English consistently group together to the exclusion of British English, and the reason for that is fairly simple. We share a more recent common ancestral population of speakers than either of us do with the Brits. Okay? So we are closer relatives, as it were. And here's just some automotive terms that nicely make the point. If I came to a gas station in the United States and said that I needed my windscreen cleaned, and you would find the stuff you needed to do that in the boot, and could you please check to make sure I have enough petrol, and so on, that would seem rather odd. So Americans and Canadians group together more frequently than we do with British English. Despite that overall pattern, however, there are certain features of Canadian English that group more closely with British English. One example on that front is the OUR spellings versus OR spellings. So Canadians have this funny habit of spelling certain words wrong, and the Brits do as well. And we do this together to the exclusion of American English. So we spell many words with OUR, and you spell them with OR. Okay, so why is this the case? Well, it turns out that OR versus OUR spellings were actually acceptably variable in pre-colonization Britain, and they were present in the early North American European colonist populations. Again, if you're interested in history, you can look at some of the founding documents of the United States, and they actually have a number of OUR spellings within them. Even though that's like anathema now, it was fine back then. Okay, so what happened here is that the OR forms were lost in Britain and Canada, and the OUR form was retained in those two populations, but lost in America. So the effect is known as incomplete lineage sorting, and not all variants in a source population sort down to every descendant population. So why is this of any use to us in understanding population sizes in the past? Well, if you can find three entities that have incomplete lineage sorting as part of them, then that gives you some information about the source population that led to those three entities. So the source population had both variants present, and also that both of those variants were present in the ancestral populations of the two most closely related entities, either languages or species. Okay. So what this basically does, it allows us to infer that a certain amount of genetic variation in species was present in common ancestral populations in the past. So if we go back to thinking about this as species, it gives us a handle on the genetic variability of those ancestral populations. So if species 1 and species 3 have the yellow, but species 2 has the blue, even though we know species 1 is more closely related to species 2 on an overall level, this gives us information that variants, the yellow and blue variants, were present in their common ancestral population, as well as in the common ancestral population of the source population for all three. So it's a way to look back in time. So what's really interesting about this is that we actually, based on other lines of evidence, had estimates of how often we should see this effect in the gorilla genome prior to our sequencing of the gorilla genome. So we actually were able to predict in advance how often we should see this effect. Now, this effect is dependent on our estimates of ancestral population size, and it's also dependent on having our species related in the right order, which we were quite confident of based on the other lines of evidence that I've shown you. So we predicted we would see this about 25% of the time with the human and gorilla genomes. The actual observation came to about 30%, which is still quite in good keeping with our estimates. And this is dependent on a population size of about 60,000 individuals. So this is now for the gorilla, human gorilla, chimpanzee, common ancestral population. We've actually also done this with the orangutan genome now, which is really quite interesting. So orangutans would be the next group down. We would have parted ways with the orangutan lineage farther back in the past. And as a result, we could expect to see some incomplete lineage sorting between the human genome and the orangutan genome. Because it's been a lot longer since we went our separate ways with orangutan, the lineage leading to orangutans, we actually predicted there would only be about 1.2% incomplete lineage sorting in this case. And we observe about 1%, which again was in very good keeping with predictions prior to that. So this gives us good confidence that our estimates of population size, as well as the time since speciation and so on, are actually in good keeping with what we're observing. So again, we have confidence that the values that we're looking at, because we can predict them in advance, are actually reliable. What's interesting here, though, just to bring the point home again, is that this measure does not depend on mutation, and it does not depend on recombination. We're simply looking at the sorting down of pre-existing variation into present-day populations. This is also why that 10,000 value is thought to be human-specific. So it seems like we didn't dip down to about 10,000 until after we had parted ways with the lineage that leads to present-day chimpanzees. Okay, so these results indicate that humans have precisely the pattern of incomplete lineage sorting that we expect, if indeed we share common ancestral populations with other great apes. So not only is this information about population dynamics over time, this pattern is also very difficult to explain unless these populations are related to one another. This is variation where it's not essential for the functioning of these organisms. Just like it's not essential that you spell it O-R and well, maybe you think it's essential that you spell O-R, but O-R versus O-U-R doesn't really matter. Either would do. But we have this particular pattern that we see, so an attempt to sort of explain this pattern from some sort of functional constraint is very, very difficult. Okay, it also shows us that the lineage leading to humans has maintained a large population size over the last 18 million years ago or more. We part ways with the orangutan lineage at about 18 million years ago. So that's why this reaches back to that point. So it also indicates that the population reduction to 10,000 individuals was probably human-specific, as I mentioned before. Okay, so far and so good, but don't we all come from mitochondrial Eve, and don't we all come from Y chromosome Adam? So you may have heard, it's actually fairly common in Christian circles, to hear that, well, wait a minute, we all come from one woman who lived in Africa about 180,000 years ago. Well, not that Answers in Genesis would say that time frame, but we all came from one woman, and we all came from one man. I think current estimates are Y chromosome, the common ancestor of Y chromosomes is at about 350,000 years ago or so. So how is this the case? Dennis, you're telling me we come from 10,000, but you're also telling us that we all come from one man and one woman? And the answer is yes. Don't worry, I'll explain. So mitochondrial DNA has a particular inheritance pattern. It's only passed on from mother to offspring. So sperm can't transmit a mitochondrion to offspring. So mitochondria are these little subcellular factories that do energy conversion for us. They have their own little circular chromosome, and they're only passed on by the mothers. So next Mother's Day, when you're writing your Mother's Day card, thanks, Mom, for the mitochondria. My wife sometimes says, yeah, that's why I'm so tired all the time. The kids got all my mitochondria. I'm like, no, it doesn't quite work that way, but anyway. I'm tired too, and I didn't pass any on. What can happen in a population? So this is the same population that we looked at previously, the same group. Because mitochondria can get stuck, as it were, they can fail to be passed on if they get into a male lineage. Like, for example, my mitochondria, they're a dead end. They're not going anywhere, right? What that means is you can have certain variants actually take over a population. So if you look at this, we've got these three different colors, red, green, and blue. And you can see that the red variant of mitochondrial DNA actually comes to dominate this population simply because the other variants end up in men, or they end up in women who don't pass it on. Similarly, Y chromosome DNA is only passed on through males. So in this example, it's the same group of individuals, but in this population, we have one individual outlined in yellow there, filled in with yellow, who is the common Y chromosome ancestor for this entire population. And the other variants of Y chromosomes have been lost because they couldn't be passed on by females, or they ended up in males that didn't have male offspring, or so they didn't get passed down to the present day. Now, regular chromosomal DNA is actually passed on equally by both genders. So there's no barrier to transmission for just regular chromosomal DNA. So what we have here is, in this one group, we have mitochondrial DNA. Everybody's mitochondria comes from that woman at the top right. Everybody's Y chromosome comes from that man highlighted in yellow. But their regular chromosomal DNA comes down from individuals, all of the individuals at the top of this group. So yes, we come from one mitochondrial Eve, who lived about 180,000 years ago, one man who lived about 350,000 years ago, plus or minus, and we come from a population of 10,000 individuals who have passed on their regular chromosomal DNA down to us from the past. Okay, why don't we sum up a bit here? So I've shown you some converging lines of evidence that support the hypothesis that life on Earth is the product of an evolutionary creation, that God used the evolutionary process to bring about biodiversity, including our own species, into being. Moreover, we have converging lines of evidence from different measures that use different assumptions that our species, as it became human, always numbered about 10,000 individuals or more. So where do we go from here? And this is where my students say, I don't want to be here. I want to come back 150 years from now when it's all worked out and I don't have to struggle with it. So I would say that a truly Christian approach needs to take both forms of revelation that we have available to us seriously. So we need to take both Scripture and science seriously, neither ignoring one or the other, nor uncritically accepting one or the other. And to that end, don't take what I say for granted. Do your homework. One way to do that is to read the book that I've written with Scott McKnight, or look at other resources. Do your best to inform yourself on these topics and try to understand why it is so many Christian biologists and biologists in general find the lines of evidence for evolution convincing. Okay, but both of these forms of revelation come from God, and we can honor God by doing the best science that we can and also the best exegesis and hermeneutics that we can. We also have to remain humble. We have to recognize that both human science and human exegesis or interpretation are fallible. Now, here's something I'll say that might be a bit provocative, but nonetheless, I'll make the point as well. In cases, however, where we have solid scientific evidence, and that evidence remains strong, perhaps over decades or centuries of examination, Christians are well within their rights, as it were. They have good warrant to re-examine their interpretation of special revelation. And the reason there's twofold. Our exegesis can be wrong. It's been wrong in the past in ways that we have no trouble with now, saying, oh yeah, it's phenomenological language about heliocentrism, no problem. So many of my students just go go past that so quickly now. I'm like, do you realize that the effort and time it took to get to that point? And now we are the beneficiaries of that particular process. But it wasn't easy at the time. It won't be easy now, but it's still something that we're called to do as Christians, to look at science and to look at Scripture. And again, if we have the conviction that God is the author of both, then we can be confident that at the end of the day that these will cohere in some way. Okay. I also see myself, my role as a believing scientist in the process is to serve the church by providing the very best scientific information possible. Now, I recognize that others who are also similarly trained don't have the same viewpoints, and that's why I'm glad that we can have a discussion here together today. But I do feel that the church needs to be well served on the science front, such that that science can effectively inform our hermeneutics and our exegesis. Okay, I'll point you at a few resources. I've written for the BioLogos Foundation for a number of years. I've been with them since 2011 in an official capacity. And there are many resources available on the BioLogos website that you could find out more about these topics on. Just a few series that I maybe would highlight to you. I've written a series called Adam, Eve, and Human Population Genetics. I wrote that about a year and a bit ago. And that's a thorough discussion of the evidence, some of which I've shown you today, but also other lines of evidence. And it's also an interaction with a number of Christians who take a different view and my interaction with their arguments. There's also the Evolution Basics series, which is on the BioLogos website, biologos.org. And that is intended to sort of be an Evolution 101 sort of series. Perhaps you haven't taken biology in a long time, but you're interested in these topics and you just want to learn what the science of evolution is all about. This is a very large series. It's 50 posts long, and it'll take you from a very simple beginning and then eventually up to a relatively sophisticated understanding of how evolution works. And lastly, of course, the book, Adam and the Genome, that I was privileged to co-author with Scott McKnight. My half of the book is the evidence for evolution and the evidence for population sizes, some of which I've shown you here, but other lines of evidence are in the book. And then Scott says, okay, if this is in fact the case scientifically, what might we think about in terms of re-examining the special revelation side of the equation? Do we have warrant to go back to the Scriptures and say, okay, maybe we've been getting this wrong? And hear me well, science never dictates what our exegesis should be. Science isn't in the driver's seat. What science can do is it can flag up for us that perhaps our exegesis hasn't been accurate. Now, that doesn't mean that science predetermines what our exegesis might find, but it can prompt us to reinvestigate the texts. So science isn't going to determine what we decide exegetically, but it can prompt us to return to the text, just like heliocentrism did in the past. And with that, I'll thank you for your attention, and we'll take our break, I believe. Thank you.
Thank you, Dr. Venema. I understood some of that, and that was quite remarkable. Very, very good, very good. All right, we're going to take a break. As he said, let's try to start back up at the top of the hour. So we'll take a break. All right, everyone. We have heard the first two presentations in the afternoon, which dealt with genetics and evolution in the broader, general discussion. The evening conversation has narrowed the focus in which we are dealing with genetics and the historical Adam. We have heard from Dr. Venema. I appreciate your presentation, Dennis, very effectively done. Now we're going to hear from Dr. Nathaniel Jeanson of Answers in Genesis, and he is going to speak to us on the issue of genetics and the historical Adam. So, Dr. Jeanson, come up, and why don't we make him welcome?
Thank you. Since it's been a couple hours since you heard from me previously, I'm going to just review real fast, and then we'll dive right into the origin of our own species. We covered the first 3 points in the previous lecture, 3 lectures ago, however long ago that was. Started very broad, asked the question, who do you trust? Because that's really, I think, what it comes down to for anyone who's lay level, and I applied that even to me. If it's a physics question, geology question, that's the same for me. I'm not a geologist, I'm not a physicist, it comes down to who I trust. We also asked the question, why are we still having this debate from the 19th century today? And I argued that it's because this question, the origin of species, of which our own species is a subset of that question is a fundamentally genetic question. And so we have not had the direct scientific evidence with which to answer it until now. That's really the fundamental reason this still exists today. We looked at the non-human species very briefly, both from a genetic and a non-genetic standpoint. I argued that the evolutionary evidences that fail to reject universal common ancestry also fail to reject the modern young earth creationist view. Now I want to focus on very specifically on the human genetic data, and if you've already gotten a copy of the book, you'll know that the chapter 10 on human genetics is organized around these 4 questions. Who do we come from? Ancestry. When did we arise? The time scale. And of course, the big question for this evening, how many people did we come from? And geography is thrown in there too. You heard some of that from Dr. Venema out of Africa. There would be a different geography Ararat, basically, Middle East, for the young earth view. I'll put the population size later. You'll see why in a moment. It's related to the first 2 questions, and actually I'm going to deal with the first 2 together because you can't separate those as well. So this is one of the remarkable facts of the modern era. I'd say maybe 20 years ago you could deal with the age of the earth and divorce it from the question of biology. It's becoming impossible to do so now. Biology is not neutral on the age of the earth. There is a clock in DNA. It records time. And this really presses the question of the age of the earth and biology together. And like I said, it's getting harder and harder to separate them. Now, we basically already covered the question of ancestry in the previous lecture. Patterns of DNA differences, shared DNA differences. Whenever we look in these categories, it becomes very difficult to separate common ancestry or to make any answer to that question of ancestry. Dr. Venema asked a question about pseudogenes, and could we use them to identify baramins? That, I think, gets the right direction. What is the function of DNA? But my answer to that is we know less than 5% of what DNA does based on gold standard genetic tests. I'd say probably less than 1%. If you use the gold standard, humans that have sections of DNA knocked out or disabled, 99% of it we haven't yet tested for function. How many of us would go to Russia if we spoke 1% of the language and tried to say, this is a functional word, this is not? I think it'd be hopeless. That's where we're at genetically. So this is an experiment in progress. The questions of ancestry are yet to be answered. That's an active area of research in young earth creation. And you think about historically, few people have actually had to answer that question. The question of where does ancestry stop and start? The species fixity folks said nothing is related, basically. It was easy. Darwin swung the pendulum the other way. Everything's related. It's only when you are in the middle here, right, basically, that you have to begin to distinguish what are the lines of evidence for common ancestry, what are the lines not. As I implied, basically, in the previous lecture, patterns, I mean, this is a question I'm interested in. I want to know what tools we can use. Patterns don't do a good job. They don't distinguish between ancestry and design. And, of course, ancestry and design are part of the young earth model. I'll explain more in a minute. It's really a question of function, and we have very little results of experiments so far that can tell us what's functional and what's not. So really, what we're going to go and where we're going to go, and this is why the time scale is so important. We're going to go to the question of what do the number of DNA differences show? Not the patterns, not the function, but how many differences separate me from you, us from the primates, primates from invertebrates. Those are the questions that we'll now use to try to answer the question of ancestry. And again, this is now very difficult to separate from the question of time. Dr. Venema already alluded to the two compartments that we'll be looking at. This is just now to put it in cellular terms. You might be able to see there on the screen there's a 1 and a 2. The 1 designates a subcompartment of the nucleus. 2 is that sort of lavender color. That's the main repository of DNA. That's the nuclear DNA, billions of letters from each parent. And then there's the mitochondrial DNA, the energy factories. And in terms of raw DNA letters, and I'll explain what the letters mean in a moment, a little basic background on genetics. There's less than 17,000 mitochondrial DNA letters in our DNA. It comes from mom. There's 3 to 6 billion in our nucleus. So it's 3 billion if you're looking at a sperm or an egg, 6 billion in the rest of our cells. So because the mitochondrial DNA is simpler and inherited in the manner that Dr. Venema described through the mothers, we're going to focus on that first. It also has implications for the nucleus, and we'll see how these two compartments interact scientifically and logically. So here's the reason why it's only inherited through moms. Again, as Dr. Venema explained, when sperm meets egg, in the oval I just circled, that DNA, billions of letters, fuses with the egg. The mitochondria are towards the sperm tail. Those never penetrate. So that's why it's only through mom. And again, there's just a summary diagram. Nuclear DNA from both parents, mitochondria only through the maternal line. Now, I'm going to use two analogies by and large for what DNA is, just so we're all on the same page. This is the diagram of the structure I showed in the previous lecture. It's this twisted ladder-like structure, so there's the analogy number one. It's like a twisted ladder, and we'll explore that a little bit further in a moment. I've also got letters assigned to it, and you might be able to see the different colors for those rungs of the ladder. So the second analogy is that DNA is like a language, and Dr. Venema explored that analogy at length. It has an alphabet. It's a chemical alphabet, and there are only 4 letters in that alphabet, A, T, G, and C. So this is the structure that gets passed on in sperm and egg. It has to be copied first into sperm and egg. That copying process is imperfect. So you can think of it, they're mutations. You can think of mutations as basically a color change in this diagram to the rungs of these ladders. Mistakes happen, copying errors happen, and because they happen, we then can compare 2 DNA sequences next to one another. And now my computer is freezing because there's supposed to be an animation. There you go. So you can see in this diagram, there's 20 rungs, 20 letters that I've compared. 4 are mismatches due to the mutations that I animated. So we would say there's 80% difference between these two sequences, 80% identity between these two sequences, 20% difference. You might say, well, how can you know you're lining up the right thing? Well, for humans, as an example, we share so much DNA, it's easy to line it up, match, 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 and every now and then a mismatch. So you can be fairly confident you're lining it up the right way. So the question is, what do the number of these DNA differences show? We can take the DNA from every one of you in this room, line it up, and get a number. And this has been done now for all sorts of people groups around the planet. We have over 10,000 nuclear DNA sequences. We have over 30,000 mitochondrial DNA sequences in our public databases from individuals around the planet. So what do these differences show? We also have mitochondrial DNA sequences from over 6,000 animal species. And over 800 mammal species. So there's all sorts of comparative analyses we can do. You can find nested hierarchies in mitochondrial DNA sequences, but once again, that fact, that pattern, doesn't distinguish between ancestry and design. So what about the number of DNA differences? This is where the time scale comes in. So evolution would say that all DNA differences ultimately are the result of mutation, this process of mistakes. And that the number of mutations in between any two species, particularly at the mitochondrial DNA level, is a product of the time scale since they last shared a common ancestor. So humans and chimpanzees, and I've shown an African woman here just because evolutionists would say the Africans evolved first. Traditionally, they put the time scale at 6 million years ago. Some would say 13 million, and I'll explain that more once we get to nuclear DNA. I'm just going to calculate it for the whole range. There are textbook equations for predicting the number of DNA differences over a certain time scale when you're dealing with mitochondrial DNA in 2 different species. It's this equation. This is really all the math you need to know for this lecture. So the number of DNA differences is a product of the mutation rate times the time, and times 2, because mutations are happening in each lineage, and we count the differences by comparing the two to each other. We know the mutation rate in humans is about 1 every 5 to 8 generations. It's been the subject of over 15 studies, highly contentious, also highly divergent in the amount of statistics each study weighted their analysis with. So anyway, you distill all that away, it's about 1 mutation every 5 to 8 generations. We don't know the rate in chimpanzees, so for a starting point, we'll just assume it's the same. When we look at nuclear DNA, we'll see that this is a pretty fair assumption. So 1 mutation every 5 to 8 generations. So think of it practically. Again, I have my mother's mitochondrial DNA, my kids have my wife's. You'd have to go back 1, 2, 3, 4, 5, 6, 7, 8 generations before you found a difference in my lineage. What would happen in 6 million years of this process in human females and our female ancestors and the chimpanzee female ancestors? Well, you can put numbers on this. And at a minimum, and actually let me back up and make one more technical point I just realized. Mutations per generation, but we're going to multiply that by years, so if you're thinking in math terms, the units don't line up. You have to convert mutations per generation to mutations per year, and the way you do that is by knowing something or assuming something about the ages at which females reproduced. Some women give birth in their teens, some women very late, maybe even 50 years old. So what we're going to do then to compensate for that range is calculate and make predictions over that entire range. And these are the results. So if all women were ages 50 in the human lineage and all chimpanzee ancestors were ages 50 in the chimpanzee lineage over the past 6 million years, there would be a minimum of 28,000 differences that are predicted between these two species. Now, I mentioned earlier the mitochondrial DNA sequence is only 16,000, 17,000 letters long. How can that be possible? What that means is you would have mutated position 1, and then 2 at some point, and 3, and all the way down to 16,569, and then you do it again. And again, that's practically what it means. If all women were ages 15 over the last 6 million years, 13 million years, you'd have over 350,000 DNA differences. That's the top of that black line on average, if they're ages 25 or so. 150,000 to 200,000 differences. So again, mutating over and over and over again, that's what it means. And you can see there on the right, the actual number of differences between our two species is only 1,400. And we'll talk about some potential explanations for this in a minute. What I want to do first is look at other evolutionary time points in our lineage. So evolutionists would say we split from Neanderthals about 400,000 to 700,000 years ago. Again, Africans evolving first in the modern human lineage. You could use a similar equation for this split. I'm going to treat the Neanderthals and humans as the same species, so you drop the 2. For technical reasons I won't get into. We can add the 2 back, but I'm trying to be generous here, and you'll see why in a moment. So how many differences should accumulate between humans and Neanderthals in 400,000 to 700,000 years? We'll again use the human mutation rate, 1 mutation rate every 5 to 8 generations. Convert that to years, depending on how old women were when they gave birth. So if all women the last 400,000, 700,000 years were 50 years old when they gave birth, nearly 1,000 DNA differences would have accumulated between Neanderthals and humans. That's the bottom of that black line. If they're all ages 15, nearly 10,000, and then on average 25, then more like 5,000. And today there's only 213 differences. So again, another overprediction, not as dramatic. We're not mutating every single letter, not every single rung of the DNA ladder, mitochondrial DNA ladder. One more time point, and then we'll talk about potential explanations. 200,000 years ago is the evolutionary origin for the modern human species, and as Dr. Venema alluded to, 50,000-ish, 75,000-ish years ago, non-African people groups split off from the African people groups. We're all part of the same species. We use this same equation that we used for Neanderthals. How many differences would we see? If all women were ages 50 over the last 200,000 years when they gave birth to those ancestors in our lineage, 477 at a minimum, bottom of that black line. Over 2,000, nearly 2,500, 3,000 if they're all ages 15. You can see an average about 1,500. In Africans, there's only about 80 differences on average. Non-Africans, about 40. And we'll talk about reasons for the differences between these two people groups in a moment. So what could be some potential explanations for this discrepancy? And these will become important, especially when we start discussing the nuclear DNA sequences. Could it be that we need to look at different levels of ancestry? Maybe we're too focused on human-chimp or human-Neanderthal. What if we go back further in time? We'll end up putting more differences in the actual column, let's say humans and orangutans, humans and a rhesus monkey. but will also add to the predicted differences. So that wouldn't solve the problem. What about the time scale? Now this is really interesting for two reasons. Actually three. The third one will arise when we discuss the nuclear DNA. What happens if you change the time scale? Well, the evolutionary time scale is interconnected, so you're going to have to begin to rewrite significant sections of the evolutionary narrative. Second reason, or maybe now up to three, third reason this is significant, is thinking more deeply about how the evolutionary time scale, specifically in geology and astronomy, is established. It's established on the assumption of fairly constant rates of change. Why do I say that? Because young Earth creation astronomers and geologists have questioned that assumption for many years. They'd say in geology, as an example, that Noah's flood, a global flood, would dramatically alter rates of erosion, deposition, so forth, that the rates we see in the present would not be a good indicator of the rates in times past. Well, now I've taken that assumption to genetics. I've assumed constant rates of change. And it gives you the opposite conclusion. So that begins to create some logical dilemmas there as well. Now, the third explanation, which is probably the most likely, is, well, what about natural selection? Could natural selection play a role? Plausibly, the answer is yes. Mitochondrial DNA encodes for some of the key proteins that function in mitochondria. Now, your heart is highly dependent on mitochondria. You're sitting here breathing, listening to me because you have functional mitochondria. So what would happen if your mitochondria got hit with a whole bunch of mutations? You might keel over and die. So that would seem a priori to be a fairly decent explanation. So we'll go with that for the moment, and we'll raise that question again when we look at DNA. But there's another question we need to ask, and I'm going to go back to Dennis Venema's book again about how you define a hypothesis and a theory. One of the defining features of a good theory is that it makes accurate predictions about the natural world. And this isn't just Dr. Venema. He's here today. We're discussing with one another. I'm using this, but you can go to evolutionary textbooks. You can look at the history of the creation-evolution debate. This is primarily the major arena of criticisms of the young Earth model, or the creationist model in general. Evolutionists have said for years, you creationists say God did it. How does that advance science? What testable predictions does that make? How does it advance our understanding of the natural world? And you can see, if you've ever done any sort of science, or you can just believe me when I say it, if you haven't. Let's say you're doing an experiment. I'm trying to understand how cancer works. I've got this drug that I think is a potential candidate. I'm going to test it in the lab. It doesn't work. Oh, God did it. How does that advance science? How does that lead us to the cure? You can see why that sort of idea would be a strong challenge to the creationist view, at least on the surface. My point in bringing it up here is the sword cuts both ways. Let's say natural selection is the explanation for the discrepancy. What testable predictions does that make? For example, one of the most divergent people groups on the planet, most mitochondrial DNA differences, are the Khoisan peoples, or the click speakers of Southwest Africa. No one has measured the mutation rate in the Khoisan peoples. What testable predictions about the mutation rate in these peoples does the hypothesis of natural selection make? That's really the challenge, not even from my own lips. That's the challenge the evolutionary community has to meet because they set it. They set that standard. And it's a good standard. It arises just out of the nature of science. Well, let me throw out another hypothesis that could explain these data. Of course, I'm going to go here because I'm a young Earth creationist. We would say all peoples go back to Adam and Eve. So we'd say in the beginning, Adam and Eve both had mitochondrial DNA. Adam is a man. He doesn't pass it on, so we go back to a mitochondrial Eve who lived 6,000 years ago. So the differences we see today should be a product of the mutation rate, one mutation rate every 5 to 8 generations, times 6,000 years. And when you do this prediction, if all women were ages 50 over the last 6,000 years, there would be around 15 differences in the global human population today. If all women were ages 15, there'd be around 80 differences. And you can see that this range of predictions, the top and bottom of that black bar, captures what we see today. About 80 differences in African people groups, about 40 in non-Africans. You can say, okay, the non-Africans, that fits maybe an age of about 25 years on average, what we kind of see today. Isn't that straining things to say that all African women would have been ages 15? Yes, and maybe not. Why? Because we have data from the United Nations. Let's say the data from 1976. Marriage data is not the same thing as age of reproduction, but there's probably going to be a correlation. So 1976 data, how old were women married? And the data now is in terms of nations, African nations, non-African nations. If you look at, let's say, ages 30 to 34, the column sort of near the middle of this, you can see the percentage of African women who were married. It was around 80%. The percentage of non-African women, about 77%. Hardly any difference. If you look at the early ages, so the top row, first column, there on the left, how many African women were married at ages 15 and 19? 33%, nearly a third, exactly a third. How many non-African women? Only about 11-10%. That's about a 2 to 3-fold difference. That's about a 2-fold difference right there. Now, that's data from 1976. That doesn't mean this has been historically true. But you're beginning to see how there might be another explanation for why there's various differences between Africans and non-Africans that have nothing to do with who originated first, but rather people group specific differences. I'll explore that question and return to it later. I want to show you this tree, though. This is several hundred human individuals. I think Dr. Venema showed you a tree also. The tips of these branches represent individuals. Way off to the right is where you'd find some of the Khoisan peoples. They have very long branches. Now, the length of each line, there are really strong parallels to a family tree here. So if you look at a family tree, the amount of difference, the distance between two peoples would be the number of generations. This is not a display of generations per se, but the number of mitochondrial DNA differences. So if you have a very long line, a lot of DNA differences. If you have a short line, hardly any. And the way you find the difference between any two people is you find the tip, let's say, at one of these Khoisan peoples, and you trace that line back to where it connects to another person, and that tells you the amount of difference. There are some things I want you to notice about this tree. So this is from various people groups around the globe. Notice how so many of these lines come together into 3 nodes, and that's not an artifact. This is a fact the evolutionary community recognizes. They call them haplogroups. This is the L, M, and N haplogroups. The second thing I want you to notice, not only is there 3 major groupings, you might say, well, there's some subgroupings. There are subgroupings, and we'll talk about them in a moment. I want you to notice the length of the lines that connect these 3 major groups. I've highlighted them here in blue. They're fairly short. So the lines connecting the groups are short. The lines radiating out from these groups are long, and we can draw an even longer one for the Africans, or I guess that direction. Let's think about this tree then from a young Earth perspective. What would be the maternal ancestry from our reading of Scripture? It would go back to Eve. Population would grow, shrink to 8 at the time of the flood, regrow again, briefly, split at the Tower of Babel into multiple people groups, and then the rest is history. So there were 2, actually you could probably say 3 major bottlenecks. So you have creation, Adam and Eve, then you have 8 people at the flood, and then the different groups at the time of the Tower of Babel. So you got short branches among the 3 major groups, longer branches radiating out. Let's think in terms of time now. You have from Genesis 5, you take those time stamps plainly for the paternal lineage. You've got about 1,700 years between Adam and the flood, but only 10 generations. They lived long. And then you see, after the flood, roughly, we'd say probably 4,200, 4,500 years. So that's about a 3-to-1 ratio almost between post-flood time and pre-flood time. And the difference gets bigger once you think in terms of generations. It'd probably be fair to say that maternal ancestry was also long pre-flood. Maternal lifespan, just like paternal lifespan in Genesis 5, would have been long pre-flood, shorter post-flood. So you'd predict that pre-flood mutations would have few opportunities, few generations. Post-flood, there have been much more generations. So what are these 3 nodes? I just gave away the answer, and I meant to build up to the punchline in a minute. So there's Eve in the beginning, 1 Eve, then the ancestry shrinks to what? You have 8 people, Noah, Mrs. Noah, 3 boys and their wives. 3 boys get their mitochondrial DNA from Mrs. Noah, and then it stops. Genesis 9, from these 3, the whole world was repopulated. We don't get our mitochondrial DNA from them, we get it from their 3 wives. So the fact that this then breaks into 3 with relative branch lengths that fit the relative time proportions seems to fit this biblical model. Short pre-flood times, those blue bars separating the 3 wives. Scripture doesn't say if they were sisters or more distantly related. And then lots of generations post-flood, those pink bars. So the structure and relative branch proportions and the absolute numbers, 3 independent lines of evidence, all fit what we see in mitochondrial DNA. Well, what about these subnodes? This model makes testable predictions. If we're all coming from those 3 nodes, and DNA differences are a marker of time, What we're looking at then, when you radiate out from those nodes, is post-flood time. In other words, the history of civilization. This model predicts you should be able to read the history of civilization off this tree. The history of the Roman Empire, the history of the Persian Empire, the history of all Greeks should be here. So what about this subnode? You see that it's coming off one of the major 3 nodes, and about halfway up it begins branching. Where do these, so the tips of these branches are individuals. Where do these individuals exist today? So let's put this together. I just hit the wrong button. Back up 1. And I didn't want to hit that, so let me go back this way. There we go. So you can see in terms of relative time, there's a branch that goes up from that node, then about halfway up that branch it splits off. So what would be half of 4,200, 4,500? It's 2,000-ish, 2,300 years ago. What happened 2,000-ish, 2,300 years ago around this section of the globe? The Roman Empire covered this section of the globe. So this is just the beginning of the sorts of studies we're doing. And in fact, if we had time, I could tell you some really electrifying results that we've been doing with the paternal ancestry, the Y chromosome. Rob Carter and I have been doing this, Creation Ministries International. We've been able to date the transatlantic slave trade. We're looking at the signature of colonialism. This is really working. It's really incredible. And that's not the only testable prediction it makes. Let's go back to the Khoisan peoples. Where are they? Way out there. I said the average number of differences is about 80 in Africans. The mutation rate has yet to be measured in some of these most divergent African people groups. The mutation rate I'm using has been measured in non-African people groups. So I would predict then, and this is a testable prediction, a falsifiable prediction, that the mutation rate will be 1 to 5 to 8 generations, perhaps faster, 1 every 2.5 to 4. So the prediction will be 1 every 2.5 to 8 generations. You can test that in the lab as soon as some Khoisan people give us their DNA. And these results, we're focusing on humans. These results are true of the other species in which we have measured mitochondrial DNA mutation rates. It's in the book chapter. I've got published papers on this. We don't have time to go into it. So why am I only showing 4 other species? You think of the history of genetics. It's only recently that we've gotten DNA sequences. It's even more challenging to get the mutation rates. So that's why there's attrition. The number of species with DNA sequences is a lot more than the number of mutation rates. This is the data that we got, and it's all pointing towards a 6,000-year time scale and challenges a millions of years time scale. So that's mitochondrial DNA. What about the nuclear DNA? Let's once again make testable predictions. The question is, what are the number of DNA differences? Patterns don't tell us much. Function is still an experiment in progress that'll take a long time to figure out. What do the number of DNA differences show? What do they tell us about our ancestry? And again, let's go with the evolutionary model first. Evolutionists would say about 6 million years ago, 4.5 to 6 million years ago, that's traditionally been the answer. Human lineage and chimpanzee lineage split, African peoples first, which is why I'm showing the African lady. BioLogos website has used the number 6 million. You can see it here, genetic evidence for evolution. So let's go with 6 million first, and there's a reason why I'm going to do this. We'll use a similar equation. That's actually the equation that the authors of the chimpanzee mutation rate paper used. So in this case now, nuclear DNA, we have the human mutation rate, which has been measured numerous times in the nucleus. That happened first, 2014. The chimpanzee mutation rate has been measured. They use this equation. How many differences should exist between humans and chimpanzees? It predicts about half of what we see. Now let me do a segue here. Dr. Venema mentioned Jeff Tompkins. We're comparing 2.2 billion of our total letters. There's total number of letters in humans and chimpanzees is over 3 billion. So where we align, so they're looking at a particular type of DNA change, SNPs, single nucleotide polymorphisms is the technical term. There are other sections you can read in Dr. Venema's book, how you get to 95%. Well, there are also whole chunks that are not found in either species. You can see that in the original chimpanzee genome paper. You can get a number of about 89% identical if you include the single letter differences, the chunk differences, and the big chunks that are missing in either case. That's been Jeff Tompkins' point all along. You see in the chimpanzee paper, 89% only matches up. You can find a match. You see in the bonobo paper, so that's sort of another chimpanzee species. You see it in the gorilla paper. You see it on the Ensemble website, one of the public repositories of nuclear information. There's only 89% that can find some sort of match. In that 89%, you can find 95, 98% identity. But the overall number of differences is upwards of 300 million if you include everything. So there's a big gap. If we focus on those to which only those sections to which this mutation rate applies, As the authors of the chimpanzee genome paper point out, 6 million differences predicts only half of what we see. So if we say, let's just focus on that 1% difference, you can only get half of that by mutation. So what's the explanation? We're not going to change ancestry. That'll run into problems. What about time scale? Well, if you bump the time scale back to 11 to 17 million years, around 13, 14 million years, you exactly capture the difference that we see. That's the option that the authors of the chimpanzee genome paper published. That was their conclusion. We should bump back the time scale. You can read it for yourself, Science Magazine, 2014. Venn is the first author. What questions does that raise? The evolutionary time scale is interconnected. So if you're going to bump back one part of the time scale, well, you're going to bump back the chimp-human lineage split from the gorilla split, and then the gorilla-chimp-human split from the orangutan split, and the great apes from the other monkeys. Or from monkeys, I should say. So chimpanzee human, if we bump that back from 6 million to 14 million, chimpanzee gorilla from 6 to 8 million to 21 million, gorilla orangutan from 12 to 16 million to 40 million, you're going to have to bump back the rhesus macaque orangutan split to 74 million years ago. Why is that significant? The evolutionary timescale puts the extinction of the dinosaurs at 65 million years ago. The first primate fossils are found around 55 million years ago. You're pushing back even this split beyond the first primate fossils. So you can see what happens when you start to rewrite the evolutionary timescale. That's what the authors went with. That's not my conclusion, that's theirs. Now, natural selection. Why am I bringing this up again? Well, let's go back to mitochondrial DNA, because these two compartments go together, nucleus and mitochondria. So the evolutionists, the authors of the chimpanzee paper are saying, let's bump the timescale back 13 million years. That means you're going to have to do the same thing in mitochondrial DNA. The minimum number of differences is not 28,000, it's now 61, 62,000. Well, natural selection can eliminate 28,000, it can eliminate 62,000. Okay, natural selection here. What about here? What about in the nucleus? What role does natural selection play? By definition, it cannot play any role in this question, because the original prediction was an underestimate. If natural selection functions to eliminate differences in mitochondria, you cannot eliminate any more differences in nucleus without making this discrepancy worse. So that raises the question, when does natural selection apply, when does it not? And I can even put it in the language of the evolutionists from 50 years ago. They say God did it, stifles science. Natural selection did it. Is that really what we're looking at now? Is that the state of evolutionary science? What creationism was accused of being 50 years ago? There's actually a fourth explanation, and I want to illustrate it from the evolutionary explanation for the origin of modern humans. So we could say, okay, our species, the modern human species, originated 200,000 years ago by the evolutionary time scale. We'd use a similar equation. Now, technically, this equation is a function of population size. I'll show you why I'm ignoring it for the moment, because it's going to be an underestimate. I'll show you in a minute. We'll use this equation to predict how many mutational differences should arise over 200,000 years, and it's only about 500,000. And today, in African peoples, in our nuclear DNA, there's about 4.3 million letters difference. In non-Africans, it's less than that, 3 to 4 million. And you only predict about 10, 12% of that. So is this an erroneous prediction? I said earlier, does evolution explain all differences by mutation? Ultimately, yes. Each generation, no. Why would I say that? Well, let's go back to this diagram and the modes of inheritance. So mitochondrial DNA is maternal. Nuclear DNA is from both parents. Now, none of us in this room are going to have parents who are clones of each other. In fact, probably everyone in this room does not have parents who are even close relatives. So your parents will have different DNA sequences. They will each pass on a DNA sequence to offspring. So that means this offspring is going to look different genetically than the parents. What percentage of those differences are due to mutations each generation? Less than 0.1%. Most of the differences you have within your body, and I'll explain what I mean more in a moment, are due to inheritance, the pre-existing variety. So that's what the evolutionists would invoke here, that our species originated 200,000 years ago, but we've been mutating back to the split with the chimpanzee lineage, and so those from, 6 million, 13 million years ago to then, those mutations were accumulating, and it's going to be a function of population size, but then those were inherited, and the vast majority of the differences between me and you are due to that and not to mutations in the last 200,000 years. So they'd say inheritance. So that raises also the question, when does that apply, when does it not? Could we apply it to the human-chimpanzee question, the chimpanzee-gorilla question? Well, now let's get to the creationist model, and this will get us then closer to the Adam and Eve question. But you see why we have to cover these questions first. And hopefully you're also able to see that the questions of ancestry and time are interlinked. Biology is not neutral on the time scale. You have to deal with it. So in 6,000 years, obviously you're going to explain just a tiny, tiny fraction of what we see today. But if evolutionists invoke inherited differences, why can't I? Now you might say, inherited from what? You're not invoking 6 million years of ancestry. So let me answer the question by asking you a question and see if you can answer it. You don't have to say it out loud. So I'm going to speak now to those who maybe would hold to a literal Adam and Eve. You read Genesis 1, God says, be fruitful and multiply. Do you think he intended for that command to be fulfilled by cloning? Well, what a weird question. Why would you ask that? Well, if you think that's weird and you'd say, no, what a strange answer. No, I don't think that's what he meant. You've basically acknowledged a particular genetic view without realizing it. So this is just a little bit more genetic background. So I've said DNA is like that twisted ladder-like structure. You can see that in the lower left-hand corner. It's also like a language, A, T, G, and C. You can see those color-coded there. Well, DNA doesn't exist in this billion rung long ladder that just kind of sticks out the end of a cell and pokes through. It's wound up and gets wound up and very tightly when cells divide, and it forms these X-like structures that are chromosomes. And this is a real diagram, picture of human chromosomes. And I want you to notice something here, and this will explain more what I was saying about 99.9% of our differences being inherited. Notice that there are some large chromosomes. Now, these are real chromosomes, so they weren't nicely electronically lined up. They're kind of floppy. These noodles is what you can think of them as. Wet noodles. And there's some small chromosomes. But notice that for every long chromosome, there's a corresponding pair, and for every small one, there's another corresponding small one. So the fact here that I want you to see is that chromosomes come in pairs. And in our DNA, there's 23 pairs of chromosomes, 46 in total. Now, you might notice in number 23 that there's a long paired with a short. That's the sex chromosomes. This is an XY individual. Y is short, X is long. This is a male. Females would be XX. Now, let's just focus on chromosome 1. It'll help us understand what's going on here in the biparental mode of inheritance. So we have pairs of chromosomes because one member of each pair comes from each parent. So you can diagram it, crudely this way. Dad contributes one member of each of the 23 pairs. Mom contributes one other member. Now, it's not like Dad contributes the information for the right arm and Mom for the left. They both contribute for the entire body, and then in a way we still don't fully understand, but it was something that Mendel observed. They interact, and your unique characteristics come out of that. So, back to the question, did God create Adam and Eve to reproduce by cloning? If your answer is yes, that means Adam had DNA sequences like this. He wouldn't have had just 23 individual chromosomes, not 23 pairs. That's lethal. So he would have had pairs like this. If you say Adam and Eve reproduced by cloning, he looks like this. If your answer is no, which is my guess is what most of you would say, that means you're proposing Adam was created with differences, or Adam and Eve were created with differences between one another. You're basically saying there were pre-existing differences created into Adam and Eve. If you say, no, they did not reproduce by cloning. That's where these pre-existing differences would come from. So, mutations contribute very little. Inherited differences contribute a lot. This is simply using a page from the evolutionary playbook in terms of the last 200,000 years into their model. I'm just putting a different number on it. What would be the signature of this? Scientific ideas make testable predictions. Well, here's, once again, the model of population growth that I described earlier. Adam and Eve, 2 people, then they grow massively. Earth gets filled with sinful people who apparently were violent, so God decides to wipe them all out at the time of the flood. Population shrinks to 8. They grow again quickly. You look at how many children each of the patriarchs had in Genesis 10. It's massive population growth, splits at Babel, and then the rest is history. Now, let me summarize a lot of population genetic theory here. In a moment. I'll use a simple illustration. So the average rate of nuclear mutations between generations is about 80. It depends on the father's age and so forth, but on average about 80. So that means there's about 80 mutations that my son has that neither me and my wife have. Now, what's the chance that his 80 rungs, 80 letter differences are going to spread around to the 7 billion people on this planet? It's virtually 0. So population genetics theory is, when the population size is large, new mutations are statistically rare. So if this is your population model, the signature of this event would be a large number of rare differences. Evolutionists acknowledge this. You can look at the evolutionary literature. They say there's an excess of rare variants in our population due to recent explosive population growth. You can actually predict this. The number of rare differences is quantifiable in our population, and it fits exactly what you'd see after 6,000 years. This is the signature of the creation flood bottlenecks. There's another signature. So if this was Adam's DNA, two different versions, even though he had no human parents, God creates him with the potential for diversity. This gets recombined. Chunks of the chromosomes get swapped. There's actually two processes. Recombination swaps big chunks. There's another process called gene conversion that sometimes just swaps single letters. So these original chromosomes will get all mixed up over time. And you can see these chunks. Scientists would refer to these as haplotype blocks. That's the technical term. You can predict how many haplotype blocks you should see in the human population today, and you see them. You see exactly what you'd expect to see from the young Earth model. Now, let me stop here for a moment. Most of what I've shown you is actually not a prediction. It's a retrodiction, taking what we see today and saying, does it fit this model, yes or no? Prediction is saying things for the future that have yet to be investigated. So I gave you some of those for mitochondrial DNA, the Khoisan people mutation rate. Reading the history of civilization off mitochondrial DNA, you should be able to do the same thing here. It's a little bit more complicated because you're dealing with a mutational process and a pre-existing inheritance process. If you're interested, I can tell you how. I'll be giving a complicated diagram in my book this fall that shows how this could give you a relative order of events. So that's the first two questions, ancestry and timescale. And I've given you some arguments for a recent origin, and I've shown you things that are consistent with coming from Adam and Eve. Now, Dr. Venema says in his book, and this is not to be contentious, but this is a dialogue. He says, to date, every genetic analysis estimating ancestral population size has agreed that we're descended from a population of thousands, not single. Now, Rob Carter has published the preliminary data that I've just shown you here as much as 6 years ago. He's published a number of papers modeling biblical population growth, very detailed simulations of population growth and crash. The data I just showed you I published last April, a year ago. So these data that I just showed you are found in that paper. So I would say it's incorrect to say that every genetic analysis agrees that we're coming from 1,000. What about the claim that many of these methods are independent of one another? That's in one sense true, but there are also some assumptions that are common to all the methods, the 3 lines of evidence that Dr. Venema showed you that are also in this book. 1, assuming that mutations are the source of all variety, and 2, they assume a long time scale, and the 3rd one, of course, assumes common ancestry. So one of the lines of evidence that I think he included in the book and has been published elsewhere is that there are too many differences to explain from 2 people over 6,000 years. That's true only if mutations are the source of all DNA differences, if you include pre-existing differences. Basically, if you say that no, God did not create Adam and Eve to reproduce by cloning, this argument goes away. He also made an argument from linkage disequilibrium, though I don't think he used that technical term, and we're all happier for it, because he makes a funny story in his book about why scientists come up with these crazy terms. So I'm going to spare you the math details and just give you the principle. It comes from basic population genetics. And pretty much all the arguments for or against an ancestral population size are statistical in nature. This is how it works. I'm going to use a silly example just to illustrate the point. Let's say you reach into a jar of jelly beans and pulling a handful out or a certain number out. I'm going to use this to simulate the process of reproduction. You might think that these jelly beans represent individuals in a population. Instead, they represent sperm and egg. So this is weird, but population genetics is weird. Sorry. So what would a bottleneck look like? You think, well, you just take one sample out and then you take 2, and you can do statistics for this. And what's the chance that both are going to be pink or both are going to be black? So in this case, sperm and egg, because we're dealing with nuclear DNA, you actually have to pull out 4 jelly beans. There's a chance, there's a chance that all 4 are pink. And I'm oversimplifying this. Normally, discussions deal with multiple generations, but the screen is only so long, so I'm going to deal with 1. There's a chance, statistically, that these are all pink. More likely, there's a chance that you'll have a not 50-50 split, and that over time, if you keep the population size small, statistically then, you'll lose variance, and eventually the population will be fairly homogeneous. Now, I'm using this as an example of one particular DNA position. You could do this for, there's millions of differences among us, so you'd have to do it for millions of colors, but that gets messy too. And it doesn't matter which color you pick, the principle is the same. You pick out a few. Statistically then, over time, your population has a strong probability of being homogeneous. And let me contrast then to a non-bottleneck, basically. So if you take a handful of jelly beans, all of them, dump them out, and you say they reproduce themselves, and this is going to be the basis for another population, well, you're almost guaranteed to not lose anything. It's a statistical argument. Again, in contrast to a bottleneck, you're going to become fairly homogeneous. So how do calculations of ancestral population size work? We're basically starting like this. We don't have the history, we just have what we see today. Is there a lot of differences, or are there a few differences? Does it look like we went through a bottleneck? Does it look like we've always been this handful, big handful, the whole jar? And that's really essentially the various lines of evidence that Dr. Venema has presented. This is basically what he's saying. We have a lot of differences. It doesn't look like we passed through a bottleneck. It fails to reject the hypothesis of a population. It does reject the hypothesis of this particular bottleneck. Well, it also fails to reject the hypothesis that God created Adam and Eve with created differences and that they grew exponentially. What happens if you have created DNA differences? And let me back up here. Again, I've shown 4 because Adam and Eve each have 2 copies. So they're going to reproduce, but they're going to reproduce a lot, be fruitful and multiply. So they're going to produce a large population of individuals, virtually guaranteeing that nothing is lost. And in technical terms, the math equations you'd use to calculate population size with the exponential population growth change. So the evolutionary literature fails to reject this hypothesis. It's essentially a straw man to say that these lines of evidence reject it. And then, of course, the last line of evidence, independent lineage sorting, assumes human-primate common ancestry. So if you assume the evolutionary ancestry, then you can make calculations that argue for population size. So I would argue, in contrast to what he claims in the book, that every genetic analysis rejects Adam and Eve only if you exclude certain explanations and hypotheses from the start. He also claims in his book, anti-evolutionary scholars have not yet mounted a convincing response to population genetics evidence, nor is it clear that they will be able to do so, since there does not appear to be anyone in the anti-evolutionary camp at present with the necessary training to properly understand the evidence, much less to offer a compelling case against it. I'll let that stand as it is. So here's the 4 questions that we've discussed, that I discussed in the book chapter. On the question of ancestry, the major arena in which the predictions will be tested is in the arena of function. Patterns do not distinguish between the various models. Function is the arena, and unfortunately we know less than, probably only about 1%. We've only done experiments on 1% of our DNA in terms of gold standard. Timescale, hopefully what you've seen in this hour, is that the young Earth model makes testable, falsifiable predictions that we can go into the lab today, tomorrow, and explore to see if they're true. For the people groups, and in fact, again, if we have more time, this hour is focused on humans, I can make predictions now for species around the planet. This isn't just limited to the origin of the human species. You've seen that the young Earth model from Adam and Eve makes testable predictions. I've made retrodictions mainly, but you can see there's predictions as well, reading the history off of our DNA sequence. And again, if we had time to look at Y chromosome, I think there's really some electrifying results that are showing up there. Hopefully what you've also seen is an indirect answer to the question of geography. Why are there differences between Africans and non-Africans? Why is there more diversity in Africans than non-Africans? So I've shown you marriage data that will affect mutation rates. What I didn't show you is a paper from 2011 looking at recombination rates in African Americans. And what they found is that the rate is faster. And the authors actually concluded in that paper saying, and I could show you the quote, that, maybe there's more to the African differences than just different size population bottlenecks out of Africa. They basically conceded there's genetic differences between these populations that may have nothing to do with their history. So I would say the argument that the evidence is consistent with a simultaneous origin of the major ethno-linguistic groups consistent with the Tower of Babel. Now, Scott McKnight says on the BioLogos website, when he was, they ran a series promoting the book, he said, Scott McKnight says, I'll put this stronger. If you don't accept Dennis Venema's section, then my section of the book need not be read. So basically, he says, we're reading Scripture after genomic science. Let's flip the argument around. In light of what I've just shown you, what can we say about Paul's arguments in Romans 5? What can we say about Paul's arguments in 1 Corinthians 15? I think it sheds new light on the subject. What's the significance of what I've just shown you? Once again, I think we've come a long way from the 1950s. The young Earth model has matured significantly. And this is, to me, just a function of time. Many, again, would point to the publication of the Genesis flood in 1961 as the beginning of the modern young Earth creationist revival. And at the beginning, you're starting it, so you're going to have few people to go along with you, and you're also taking on a massive scientific enterprise. So the posture the early young Earth creationists took was largely defensive. It's just the natural consequences. Evolutionists would say this, and young Earth would say, well, not so fast. Well, what about this? And here are some problems. Fast forward to today, I would be a second generation, if not a third generation young Earth creationist. We've got a large community of scientists who work on things together. On top of that, we finally got access to the genetic data, the most important scientific data in this debate, and it is freely available. So people can you can sit at your laptop, if you know the right tools, and do your own analysis, because all this data is out there. So you've got a bigger community, a whole lot more data that's freely available. This evens the playing field and allows young Earth scientists to make testable predictions. And I would say that actually exceed the testable predictions, the ability of the evolutionary model. And it exceeds it, and this is, I think, really the kicker, on the most important arena of genetics. What is the mechanism of evolution? It is the mutation rate. It's mutations over time. That's the ultimate explanation. And it is the young Earth model that is taking the lead in this field, the most fundamental aspect, the mechanism of the evolutionary model. So Dr. Venema, again, has a fantastic intro in his first chapter, hypothesis that is not rejected. After many predictions and tests, becomes a broad explanatory framework, makes accurate predictions about the real world, a theory. He would say evolution fits that definition. I would say young Earth creation fits that definition. It remains a productive explanatory framework. Hopefully that's what you've seen this hour. Not only has it been failed to be rejected over 50 years, it remains a very productive and fruitful explanatory framework. We're working on the research questions right now. I think it's extremely exciting. Anyway, you can see my excitement. Question for you, of course, is, bigger picture. This is one small drop in a very bigger debate. Who do you trust? That's going to be the question. And whoever you trust, how do you explain the other side? So, hopefully what you've seen explains for you then why I'm titling my book Replacing Darwin, because I think we've gone beyond rebutting Darwin. That was the defensive strategy of decades ago. Now we've advanced so quickly, I think we can actually replace Darwin, and roles have now been reversed. That's what I'd argue, and will be arguing in this book. You can get the book chapter in chapter 10 of Searching for Adam is available on our table. We actually have 4 books out there, normally retail for $72. You can get them for $20, so it's a real saver. Again, we didn't have time to discuss the bigger general question, how do you get tens of thousands of species from just a few on the ark in a few thousand years? That's what I cover in this lay article series and much more in depth in my technical papers. So, thank you so much again for the invitation, for the opportunity to present to you, and for staying on this late evening.
Thank you, Dr. Jeanson, for this informative and stimulating presentation. Very helpful. We are going to take a brief break and then start up once again at the top of the hour. And at the top of the hour, we will then move into the conversation between the 2 speakers, in which they're going to interact with what they've heard one another say. And then we will take the questions that you have been presenting. If you haven't presented your question yet, do so on the Facebook page for Sebets. All right, let's take a brief break. everyone. This has been, this conference has exceeded my hopes and expectations. I have heard both men speak in other venues, and I knew that they were both very bright, very articulate, but both of you even surpassed what I had hoped and expected. It was quite remarkable, your presentations. As a non-specialist, I'm always blown away by what you have to say, but not just that, just the way that you are able to take some really difficult concepts and are able to communicate those as well as you have. I want to apologize to the audience who have been watching over Facebook, the streaming video across Facebook. I think we've had over 10,000 different viewers this afternoon, so we've had a spectacular audience. The difficulty is that they've not been able to see the animation in the PowerPoint, which has put them at a tremendous disadvantage. The good news is that when we put the material on the CFC website, we will combine the respective PowerPoint presentations with the audio and video talk, and at that time, one will be able to see it. Of course, that'll be too late to help the students who are writing the reports because it'll take them a month to do that. So you all, unfortunately, are out of luck. You'll have to write it as it is. Dr. Venema, you just heard Dr. Jeanson and make his presentation. Let me give you the first opportunity to give your observations of what you've heard and perhaps questions you might have for Dr. Jeanson. And then after he does so, then I'll ask you the same question.
Okay, great. Am I on then?
Yes, you are.
Now I'm on. Good. First off, I want to say thank you to Nathaniel for your presentations. Very clear and articulate, and I appreciate them. Excuse me. I also want to underscore the fact that I think it's okay for brothers in Christ to have these differences and have these dialogues. So I hope that this can be a model for how to talk graciously about these issues. I recognize that for some communities it's seen as more of a central faith issue than for other communities, but I think even, like Answers in Genesis says that this is not a salvation issue, right? This is an issue, one's interpretation of the early chapters of Genesis does not.
You'll have to ask him.
Okay, we'll let that go. I think I've read that on the website.
Is that question one?
Just right off the top, I want to read something in my defense from the book that talk about where I say, there aren't young Earth creationists that are correctly trained in population genetics. And that's actually something that doesn't come from me. That actually comes from within the young Earth creationist community. So we've mentioned Todd Wood before. This is what Todd Wood has to say about it. So I'm basically just taking what he said and sort of rephrasing it. So Todd says this: the population reconstructions are complex and not easily understood by lay people right now. So creationist responses lag behind the current science, and the best your typical creationist can do is cast aspersion on the science. Until we have a creationist well-trained in modern theoretical population genetics, I think we will continue to have only unsatisfactory answers to these ancestral population reconstructions. So I guess maybe one comment to say right off the bat is that this is not just a conversation between young Earth creationists and evolutionary creationists. It's also a conversation within the scholarly young Earth creationist community. And one of the things that I've enjoyed over the years, I actually have a friendship with Todd. I haven't yet met him in person, but we email back and forth. To be completely honest, Todd says, Dennis's advocation of evolution makes me very uncomfortable. And that's reasonable. But he says, but we have a lot of common. And he says, we have Jesus in common. And Jesus, he says, is a lot to have in common. I just love that particular turn of phrase. So yes, Jesus is a lot to have in common. Absolutely. So that was just on that particular point. I guess what I'll be looking for in the coming years is, Todd is somebody who has read Carter. He has read Tompkins and whatnot. So
That's actually incorrect. So it's not actually a conversation within the young earth literature, and I don't mean to be belligerent. He's a unique character. Jeff actually worked with him for many years, I think, at Clemson. And he has said, it's a real shame. Todd's really talented. He's reached out to Todd Wood. I've reached out to him, say, review my papers, and has refused. To me, conversation is a two-way street. You talk, you listen. I'll read his papers. I'll cite his papers. Jeff has reached out to him. Can you please review this? And he refuses to engage any of this. And so, to me, his statement saying, there's no young earth creationists, I'm like, he also doesn't seem to be aware of Rob Carter's work. And so I know from my own personal interaction, he has declined to engage what we've published and shuts himself off. And he said on his blog, he says, what I really wish, my burden for the young earth creationist movement is to have an independent voice, sort of an independent critic. And that's kind of, you read his blog, and that's kind of how he styles himself as. He says he's too tied to the young earth creation movement to be that, but actually he's distanced himself very significantly. That's not something we go around parading, because we're not here to bash people, but that is the behind-the-scenes reality that we haven't made public just because we're not interested in bashing. But sadly, he has not shown any interest in conversing about it, but seems to feel no restraint about posting publicly things that are not true. So he's not actually having a conversation. He's one of these splinter groups that has their own following. You have to ask anytime someone says young earth creationist, well, what's their means of accountability? You'll see things like on our website, other websites, arguments we think young earth creationists shouldn't use, and you'll find out the young earth creationists are quite offended because that's their favorite argument, and even creationist organizations, well, how dare you say that? And people come alongside, and I have to consider this data. They don't want to be accountable, and so, and we seek accountability. Like I said, at Ancestry and Genesis, we have an editorial review board that reviews everything that goes out. We seek outside review, independent review. We'll have sometimes bi-yearly meetings on subjects. We had one on the Ice Age, because there might be different views, and one exactly within the young earth framework that works. We try to make it a continually self-correcting process, whereas Todd seems to have removed himself from that, not just that process and accountability, but doesn't even want to even talk about it, which is really sad, because he's a very talented individual. But as an example, he published an abstract. So not only has he not done it behind the scenes, he's not done it publicly. To me, the best way for accountability is written peer review. You've probably experienced this as well. I could tell you other stories about how I've experienced it. People are resistant. And that peer review comes from the nature of science we discussed, as you laid out in the first chapter. Science is an inductive method of reasoning. You never prove anything in science. You can only disprove. We're all fallen human beings. There are things I may have missed. There are things Dr. Venema may have missed. And you seek other scientists to say, you know this methodology. What hypothesis have I not excluded to jump to my own? And in hopes that the groupthink will then arrive at the conclusion. So that's what we seek to do. And anyway, so a lot of what Todd has published, let's say on human-chimpanzee genetic similarity, he published something in 2006. Most of Jeff Tompkins' work postdates that. Todd has published a few abstracts, which are maybe a paragraph, which, if you've ever read the scientific literature, that's not something you can critique. It doesn't give you methodology that you can repeat. It doesn't give you a way to critique it. So he really hasn't published anything, either on the topic, that we can evaluate. Where Jeff's methods are transparent. He's got detailed lists. So unfortunately, he takes the label, but again, it's not a copyrighted label, and he resists accountability on that front, and even resists our invitations. He sets himself up as critical, and I ask, my boss, hey, can Todd review these papers? And each time, no thanks. Just takes a very negative attitude, which is a shame.
Well, obviously, I don't know the ins and outs of young earth creationist sort of behind-the-scenes sorts of things. What I find interesting about Todd is that he's trained. He's a geneticist and has good training. And in my interactions with him, I've found him to be approachable and willing to dialogue. And yeah, I enjoy very much hearing what he has to say. So yeah. Let's see, what else should I ask you about? I'll fully admit to not being completely up to date on young earth creationist baraminology. what does Answers in Genesis hold as the human baramin at this point? And I know Todd is an outlier on this point as well, because he includes an Australopithecine within the human baramin. What's Answers in Genesis' take on that right now? How far does the baramin extend?
And just a quick clarification, baramin is sort of the colloquial term.
Created kind. Yeah.
Baramine, yeah. Not colloquial, the technical term. That's why it's colloquial if we understood it all.
Yeah.
So
my latest understanding is that like Neanderthals are human, Denisovans are human. I'm just not sure where Answers in Genesis is at on that front.
And so there's a history to this field of baraminology. That's been sort of the term in the young earth literature. I tend not to use it as much just because it tends to be a very specific meaning. But the basic idea is something I referred to earlier, which is we're not species fixity proponents. Again, you read Scripture from our perspective. God creates kinds, not species. That's not the term used. And on top of that, the land, everything kinds are wiped out, restart in the Middle East. So it's kind of beyond me why the creationists of 1859 would somehow think that geography was fixed when you have a geography that's totally rewritten from a young earth perspective about 4,500 years ago. So we would say there's limited common ancestry. Scripture, again, doesn't give you a whole list of terms. I'll give the derivation very briefly here, and again, you can read more of it in our literature. So Noah is commanded to take two of every land, every breathing kind, male and female, for the purpose of propagating the seed or the offspring. So that then implies, the purpose is reproduction. Kinds must be able to reproduce. And so as a crude first approximation of what might a kind be then, we've done breeding tests. And since this is dependent upon a specific scientific test, where that kind boundary lies is going to change with time with the data that we have. So 50 years ago, you might not have very many breeding tests, and they might say maybe it's genus. And then, we can actually connect to almost every cat species in a reproductive continuum. Now we're going to bump it up to the level of family. As we do more breeding tests, might that change too? And really, to me, the ultimate answer is going to, again, be genetics. Fossils aren't inherited. Geography is not inherited. DNA is inherited. So that's going to be the record of ancestry. And it's going to go back to these questions of, it's going to go back to the standard of science. We're going to determine what the kind boundary is by seeing which explanations make the best testable predictions. If I say it's the genus and I can't predict anything in terms of genetic diversity, that's probably not a good hypothesis. If I say it's family and it's starting to explain what we see, that's the process we'd go about doing it. So then the challenge is, back to your question, what do you do with fossils? So I didn't mention the Neanderthal DNA when I was talking about 6,000 years for a variety of technical reasons. I think the question is still open. Most of my evolutionary colleagues would say that they think the DNA is reliable. They would all acknowledge that it's degrades from the ends. You're talking about stuff that has been sitting in the ground from a young earth perspective for thousands of years, from an evolutionary perspective for millions of years. When I was doing DNA analysis as an undergraduate, we would stick stuff in the freezer, minus 20 degrees Celsius for a year. DNA would start doing weird things after a year, and we just toss it. So I'm highly skeptical, just based on that experience, that we're ever going to be able to recover some sort of reliable DNA from Neanderthals. And I've made technical arguments in my paper saying it doesn't even fit the structure. If anything, if you're going to say it's reliable from a young earth perspective, you have to say it's some sort of hypermutatable. I think, again, the most likely explanation and most boring explanation is that it's degraded. So to me, we lack the tools. Todd has been trying to do a method where he tries to quantize the number of non-genetic features among various creatures or species, and seeing if you can find groups. Now, what's the challenge with that? Number one, it's not genetic data. Number two, it's highly dependent on which features you pick. And that's been some of the discussion in the young earth literature, some of the responses to his work. Well, you're only looking at teeth and this. And actually, I asked Dave Menton, who taught anatomy and physiology at Washington University for like 33 years. So what do you think of Todd's method? He's like, well, it's exactly what I'd expect from a geneticist trying to do anatomical comparisons, basically, which is, that's not how you do it. So I think the jury is still open, I guess is a long way of saying that, because I think we lack the good tools by which to analyze it. I think it would probably be fairly unanimous that Australopithecines would not, going with what the criteria Dave Menton would use, saying here are the distinguishing features of the species we see today, chimpanzees versus humans. And in many cases, the fossils that are prominent and being debated may not even have fossil data for that particular distinguishing feature. Certain let's say you're talking the skull, a certain bone up here in the nose, or you look at the fingers, or anyway, Dave Menton can speak that a lot better than I can, but that's where it's at the moment, that Todd has been using largely his particular methodology, which is statistical quantitative, highly dependent on which data you're using, and you can see that in some of the history of his results, and not necessarily weighing distinguishing anatomical features more heavily than things that don't really distinguish species.
Okay, good. Okay, another question. One thing as a biologist that I'm often curious about with the young Earth literature is that many baramins, or accepted groups of relatedness, have DNA diversity within them that are actually far greater than human and even human and chimpanzee differences.
Mm-hmm.
So I'm curious why that is the case, or how that fits in a young Earth creationist model. If we can see diversity in related groups that exceeds the human-chimp difference, and from a young Earth perspective, that's happening in a very short period of time, then why, how is it that we can baraminologically, reliably locate humans as not related to other species?
So the question basically is, if you look at nuclear DNA sequences, there's a whole bunch of ways you can compare it. There's different categories of differences. Single nucleotide polymorphisms would be single rung differences, basically, where I have an A and you have a T. Indels, insertions and deletions would be, I've got a row of these 3 rungs and you're just missing those 3. You've got whole chunks. It might be, I've got this section of 3 million rungs or 500,000 rungs and you're missing it altogether. These are the sorts of things you'll see. We've got 4 to 4.3 million or so, 3 to 4 million single rung differences around the globe, and then a variable number of these other types of differences. Actually, the indels are more like, the small ones are 600,000 or so. All that background to say, let's compare species at the single letter differences. It's easier. And we have data for that, and it's data that I remember offhand, so it's even better. So for humans versus chimpanzees, the single letter differences in the regions you can align, again, ignoring that part that doesn't align, you'd have around, well, you saw it, about 26 million or so, 1% difference. Let's say you compare the tiger to a house cat, which we would say are part of the same kind. There are almost 50 million single letter differences. As a general rule of thumb, we'd say that species within a family have a common ancestor. So the great apes belong to a single family. Evolutionists would put humans in the same family. We wouldn't, but chimpanzee, bonobo, the 2 orangutan species, 2 gorilla species, all part of the same kind, we'd say, came from a common ancestor on board the Ark. The orangutan, gorilla, orangutan, chimpanzee differences, I think, anyway, they are 3.4% different. I think it comes out to around 100 million differences. So 10 times, well, maybe 5 times higher than what you see in humans. So what does that imply about ancestry? And so this goes back to the question then of what are the tools we're using to determine it? And are there patterns? Are there numbers of DNA differences? And none of those criteria seem to be good tests. Ultimately, the test is what testable prediction does a model make. So right now, I would say it would seem that God created, let me back up. The data I didn't show you, some of which is in my article series and my technical papers, is young Earth creationists like myself. I've argued that this idea of pre-existing diversity, God creating Adam and Eve with differences within themselves, applies universally among species. That the cat ancestor on board the Ark was front-loaded with a whole bunch of different DNA differences, and those then have led to the origin of species. Actually, the primary cause, we'd say, the origin of species. So from that perspective, I would say it would appear that God created animals with more potential for diversity than humans. So as it relates to the question of baraminology, for many years, the criterion has simply been breeding. And then now, because we're getting DNA data, we can finally bring genetics into it. And so we're now having to develop criterion. What sort of tests can we use to identify common ancestry? Well, what testable predictions does a model make? So if I can say, let's use mitochondrial DNA. Let me think of actually a better example. Bovids. So Bovidae family, 135 species alive today. Everything from cattle to sheep to goats to African antelopes. Very diverse family. Cows and sheep differ by 10% of their DNA, I think. So 10 times higher than human chimpanzees. We'd say maybe they have a common ancestor. They're clean animals, so it might get a little sticky, but that's a side note. Mitochondrial DNA, that's where I was going with this. So there's maybe 2,000 differences between cattle and sheep at the mitochondrial DNA level. Humans versus chimpanzee, there's only 1,400. Humans versus humans, you saw the data, 80 to 40. So how do you explain the fact that you have cattle and sheep potentially sharing a common ancestor, but there are thousands of differences apart? Humans, we'd say, have a common ancestor and not related to anything else, less than 100. That'll probably be a function of DNA differences, mutation rates. So no one's measured the mutation rate in these bovid species. We can make testable predictions, and really that's what it's going to come down to. So I would say we've made retrodictions for humans. We can make testable predictions for Khoisan peoples, other people groups in which it is yet to be measured. I can make predictions then for sheep and cattle, and the success of this model will be seen on how well those predictions bear out in reality.
As a lay person listening to you, I just want to make sure if I understood what you're saying. Are you saying that on the ark there was a common ancestor for sheep and for cattle, and that
potentially?
Okay, you are. What's potentially mean? Just so I'll understand. Are you saying then?
For sake of argument, yes, but because they're clean, and then there's the question of 7 of the clean, 2 of the unclean. Could it have been that there was 3 different looking representatives? I don't know. So for unclean, cats would be an easier example. Two cats, and you have everything from tigers to house cats to jaguars in between.
For my simple mind, sheep and cattle are pretty dramatically different species. Mm-hmm. So whenever I hear someone saying that's a common that they had a common ancestor, well, I got in trouble for everything I thought about that. I thought that was quite a remarkable thing. So I just wanted to make sure I understood that. Do you have any other questions you want to ask him before I turn him loose on you?
Sure, sounds good. Just maybe 1 or 2 more. If
okay.
That's okay. Then it'll be my turn, right? Then I'll be on the hot seat.
Yeah.
Just a quick question. When you say those examples of pseudogenes with mutations and nested hierarchies, do young Earth creationists accept that within accepted baramins? Like, would you look at gorillas and chimpanzees and orangutans and say, yeah, they would have inherited that mutation from a common ancestor?
I would like to.
Okay.
So it goes back to the question of how do you identify common ancestry? And if there was a mutational signature, I would embrace it quickly. But I would say what we call a mutation and what we don't is virtually impossible at present because of the non-intuitiveness of DNA. So again, back to the language analogy, we speak less than 1% of the DNA language, as far as I'm concerned. You just have to look at the history of genetics. Back in the 40s, 1 gene, 1 protein, 1 function. I'll talk to people from a generation ago. I taught a class at Southern Evangelical Seminary on biology and origins, and we had a gentleman there who's like, everything I'm learning is the opposite, or this is totally different from what I learned in school. It's changing that fast. And so what you might look at a textbook a decade ago is now being totally overturned. And so things that look like mutations, because they look like broken genes, I think of pseudogenes as a specific example. So when I was in graduate school, one of the newfangled, unexpected ways of DNA function is actually something near and dear to my heart. So what I worked on as an undergraduate is RNA. So DNA is transcribed RNA translated to protein. It's the intermediate between the two. We're working on RNA regulation. My boss was always, RNA is the underappreciated, this is the big deal, and we need to make more of a stink about it. Well, he's basically been vindicated because the amount of literature on RNA function is more than I can tally. I mean, there's reviews written on this. Anyway, back to graduate school. So one of the shocking discoveries, I think made originally in plants, was RNAi, RNA interference, small, short RNA snippets that bind to, and I think there's base pair differences there, they look like mistakes, bind to other RNAs and regulate the function. And I remember sitting in class and one of my profs saying, Andrew Fire was in Massachusetts. He was one of the guys, he's going to get the Nobel Prize, and sure enough, got the Nobel Prize for this. So what are pseudogenes? They're sometimes antisense versions. To me, what comes to mind immediately is they're going to function in some sort of RNA regulation. It's just too close. Does it look like a broken gene? Yes, but to me that's decades ago biology. And I'm not being insulting. My point is, I don't disagree that it looks like a broken gene, but I also see this massive literature that's saying there are all these unexpected things RNA do. And then I look at the fact that we've actually functionally tested less than 1% of the genome. To me, the genetics community has done a really bad job making predictions. in terms of function. And that's not an intelligence thing. It's just because we're dealing with a foreign language, a highly compressed. It's beyond the best computer code, is really what it comes down to. I mean, we write books. We don't write books that can be read forwards and backwards. DNA can. You've got proteins. There's a phenomenon, and I've written part of this in the paper, one of the first papers I wrote, protein moonlighting. So one of the 1940s ideas was one gene, one protein, one function. So some of the mitochondrial DNA proteins. Well, we know the function. It functions this particular enzyme complex. It takes this chemical, transforms it to this, releases water, whatever, transforms oxygen. This particular enzyme takes glucose and it removes this group, and it's at this step. This is classic. It's known. Well, now we're discovering serendipitous things like, oh, this enzyme that functions in sugar metabolism is now binding to DNA in the nucleus. Anyway, moonlighting. Proteins are doing things that you don't even anticipate. Let me explain it by analogy. Proteins, if you think of our bodies as biological construction projects, we start as a single cell, and all of this is built in the womb, basically, so that the human form we recognize as this is apparent by birth. Proteins are one of the main tools for building the body. Protein moonlighting is analogous to having a multifunctional tool, but what human engineer builds a handle that is a hammer, saw, drill, and a whole bunch, 10 other things in one hand. I mean, we don't even design. It's science fiction. What's going on at the cellular level? So
if I'm understanding you correctly, if I could just jump in. You're saying that the DNA code works as if I were reading a book. If I read it this way, it would be a poem, a Shakespearean sonnet. If I read it the other direction, it would be perhaps a newspaper clipping from the Washington Post, and it works both ways.
And so again, some of the classic paradigms, you can go to some of the textbooks I have, they'll say, even in those things we thought we understood, let's say this particular DNA sequence codes, is transcribed to this RNA, which is translated to this protein. There's this talk of synonymous codons. So in short, there's 20 amino acids, 4 different DNA letters. There's different languages in your cell. And 4 different DNA letters can't code for, one letter then can't code for all 20. It's groups of 3. 3 DNA letters code for a single unit of a protein. And I was taught the third, so there's some redundancy. That's the point I'm trying to get at. There's some redundancy in DNA coding for protein. Well, I was taught, there's this wobble position 3. If you look at textbooks, statistically, then, it can be variable. Well, now we're finding that actually it plays a functional role. And there's even reviews being written about this now, that these aren't just, it tolerates error. The cell uses different redundant elements to slow and speed the process. I mean, the level of information compression into every single letter is really, it boggles the mind. So that was a different example than you're asking about, but it's a crazy field.
One more question?
One more question.
Okay. Although there's some things I would say in response.
Yeah.
Please do. We can have a dialogue.
No, this is a dialogue.
Oh, okay. Well, I don't want to take up too much time, but there are cases where we see pseudogenes, where we actually see many cases actually, where we see the same gene in a functional state, say in mouse, relative to the human genome. And we have the ability to determine its function experimentally in mouse. So one of the examples I gave was along those lines. So what do you think of those kind of pseudogenes, like where we actually have experimental evidence that it has a certain role in mouse, it's in the same little block of genes in the human genome, yet it's non-functional, or at least it can't have the same function that's in the mouse. So why is it that we see that pattern? I'm curious, from a young Earth perspective, what does one do with that?
I would say, and you can tell me if I'm articulating the argument correctly, there's an element of homology in the argument, and there's an element of non-function. You have the homology in that it's shared, but then the other element of the argument in human looks like it's non-functional.
Sure.
So to the homology element, I go back to design and saying human engineers reuse common patterns for similar purposes. What would be the purposes then in DNA? And that's, to me, where it gets sticky, because we're so bad at determining purpose. I mean, the whole idea of protein moonlighting, the whole idea that RNA is doing all these crazy things, and is it weekly, monthly, that some new discovery is coming out? So can I predict at the moment what that human gene is doing? No, but in 5 years, I have a pretty good guess that I will. because it's going that fast. And it's not what we anticipate it to be. And there's so many interactions in the cell. So one of the hottest fields right now is the 3D arrangement of DNA in the nucleus. That the cell not only uses DNA according to sequences that exist before and after the gene, but it's how it's compressed and uncompressed and wound together. There are so many serendipitous discovery, I think it'd be foolish to try to assign a function. I don't know that anyone could assign a function, and what it turns out to be will probably blow our minds. And I think it would be highly premature to say it's non-functional because of the trajectory. So many paradigms have been overturned in the last decade. I mean, I graduated 2003 from undergrad, and I have my undergrad textbook. And so, what is that, 15 years or so, and things are being overturned then. It's so fast. Like I said, I would love to have a tool to say, this shows the signature of Ancestry, this one doesn't. And I don't think I can do that, given how fast things have changed in terms of what we say is functional and what we say is not.
One last question. I asked this question of Georgia Purdom back in 2011, I think it was. She and I had a similar dialogue at LeTourneau University, and it's out there on YouTube if you want to see it. I asked her this question, and I'm curious, so I remember her response, and I'm curious of your response as well.
See if we're consistent.
See if you're consistent or not. Not that I'm trying to catch you out or anything.
I'll keep my phone in my pocket in case she's watching and
okay,
going to text
sounds
me.
good,
We
yeah.
can't cheat.
Yeah, and I may not get it exactly the same, but I asked her something to the effect of, do you think, is there any evidence that could potentially convince you that your position on origins is incorrect? Is there any, can you conceive of evidence that might convince you that you've taken the wrong tack on things? I'm just genuinely curious.
It's a good question. And I would say it's more of a philosophical question.
Yeah, in some ways, sure.
There's a scientific element, a theological element, and a philosophical element, and I'll answer each of those in turn. So, the scientific element, I've given falsifiable predictions, and I'm happy to go test in the lab. Let's go do it. Let's go test this. What have I been saying all along, though? Take evolution versus creation as an example. The non-evolution, the non-genetic evidences. I've said, this fails to reject evolution, fails to reject young Earth. There's all sorts of hypotheses that it probably fails to reject. Within the young Earth model, we'd say there's, Scripture clearly teaches these things, and on these things it's silent. And even in the arenas where it's silent, there's a whole lot of hypotheses you can create. So, let's say I make a prediction on the mutation rate and it's false. Does that mean it's only this or it's only this young Earth position or not? There's probably 5 other young Earth positions that fit it as well that it could create. So basically what I'm getting at is science is it's simultaneously a very poor and very powerful tool to know the world. It's very powerful because we're sitting here with our iPhones and technology is advanced, and that's due to science. But it's very poor, especially when it comes to origins, because the further back you go in time, the less and less you know about what potential competing explanations exist. And so, in that sense, science is very weak. And I think it would be foolish to base such a gigantic question such as origins and the truth of Scripture on such a weak and unstable discipline. Powerful discipline, but inherently weak in terms of absolute truth, and such big, big questions that touch on so many topics. So religiously, we have a very specific hermeneutic that we adhere to, so it would require hermeneutical change to change my view then on Scripture. That's tied then to philosophical as well. So what is my view of origins? Origins is a multidisciplinary question in which science plays a role, a limited role, because all it can do is eliminate competing explanations, and there's a whole lot of them that need to be eliminated. And science is inherently pragmatic. At the end of the day, there are some, and I would like to consider myself one of them, who are interested in knowledge for knowledge's sake. But a lot of technology is driven by, does it work? Can it work? Great, move on. We want to send men to the moon, so we're going to do the physics until we get it right. Do we need to do the physics until we understand the last detail about the nature of matter? No. We've got men to the moon. It's going to get funded. I mean, there's a money roll in all this too. So that also makes science a weak way to answer big questions. It's a great way to know the natural world, but when we're dealing with natural, supernatural, and all that, you can see how this becomes weak. I love science, and I love to do science, and I think it's super exciting, the research I'm doing, but I also recognize the inherent limitations and the need to bring in Scripture and other things as well. So I fully embrace making testable predictions, recognizing that there's a whole lot more. And you being an old earth creationist, I was fully anticipating you, the wheels turning and saying, well, how does this work with old earth creation? I'd say, well, there's probably testable predictions I could come up with there too. Just as an example, a practical example. Does what I show you tonight distinguish or eliminate old earth creation? Fuzz Ron and you, Ross, would have to answer that. And it might not. There might be some things I've shown you tonight that might seem to reject evolution, but not reject old earth creation. You see where I'm going with this. The point is, I haven't considered everything that's out there. And probably neither Dr. Venema or I will ever consider everything that's out there. That's why science changes so much as our knowledge of the world grows. So scientifically, do I have hypotheses that can be falsified and changed? Sure. Will that change my young earth view? It's going to be pretty tough because there's so many things that are still compatible with a young earth view, other hypotheses on specific topics. And to me, that's true in general of science. I mean, even hopefully, as you've seen tonight, when it comes to potential evolutionary explanations for the different compartments, you have inherited differences, you have selection you can deal with the time scale. I think you'd agree. Even with any evolutionary framework, it's hard to find an either or. It's either this element or this, and this, versus either this, and this, and can we find one where there's what I call in the book a category 1 experiment, where it's going to, if it goes this way, it's clearly young earth, if it's this way, it's evolution. Oftentimes, most of the time, it's never the case. It kind of favors this way, but over this way. So I think that's the reality of what we're dealing with. And so scientifically, falsifiable predictions, yes. Religiously, it's a hermeneutical question, and that's entrenched in philosophy as well. So,
well, I am glad that you pointed out that I'm actually in a mediating position. And that's one of the reasons I have enjoyed, one of the many reasons I've enjoyed this night is that I'm actually in a position literally in between the two of you. And so, so that has put me in the proverbial catbird seat in more ways than one. Dr. Jeanson, excellent presentations, excellent answers. Now it's your opportunity to make observations and ask questions to Dr. Venema.
So first of all, thank you as well. And hopefully you've seen, I've really enjoyed, especially that first chapter and the way Nature of Science laid out. And even many, as you're presenting the language change and such, sitting there thinking about speciation in 4,500 years, and that's a useful analogy, and here's how we see DNA change. And actually, if you read, anyway, I'm supposed to ask you questions, so I'll stop and ask you questions. So I presented some strong challenges on mutation rates and such. What would your response be to those, or some of the things I put more punchy?
Sure, yeah, sure. Certainly on the variation that we see in mitochondrial DNA. Mitochondrial DNA is very constrained, right, because it's very small, and there's very little, as you know, as a biologist, there's very little space in between genes on mitochondria. And in fact, many of the genes actually overlap one another. So I wouldn't expect, and I don't suppose you would either, just as a biologist, that the differences, the raw differences that we would see between different groups would solely be the result of raw mutation without any selection that's actually removing deleterious mutations from the population. So I wouldn't expect to see those raw differences merely just be like a sort of a neutral clock of certain mutations. I was actually curious about your thoughts on Neanderthal DNA for that specific reason, because if you include Neanderthal mitochondrial DNA, then of course that makes the problem. There's a lot more variation there to account for. So I would see definitely selection for that particular limited, because of the sort of dense nature of the information that you see in a mitochondrian's genome. When it comes to nuclear DNA, I would be more comfortable with thinking about it in more neutral terms, because the evidence we have is that a large amount of our DNA doesn't have a sequence-specific function. So I'm more comfortable as sort of a neutral, without considering the effects of selection more generally for nuclear DNA for that reason, which is not to say that there isn't strong natural selection on certain parts of nuclear DNA, absolutely. So that would be my general thoughts on that particular question.
So I'll put it more punchy.
Sure.
Because I did it in general to an audience. I'll put it to you. So what testable predictions would you be able to make from that mindset? Can you predict the rate of mitochondrial mutation in Khoisan? Can you predict the rate of mutations in cats?
Sure. It really depends on having an understanding of the functional constraints on that particular one. So again, it's a lot easier to do that kind of analysis when you're looking at sequences that are almost neutral. And one of the things that we can see in nuclear DNA is you can actually look at nucleotide substitution rates when you compare related organisms. And we see that nucleotide substitution rates are actually much larger or much faster in regions that don't correlate with things that we think are functional. So if you look at gene coding sequences, they have a substitution rate that's far below what we would see in, say, introns, the bits in between coding sequences that are spliced out. Now, there's still some functional constraint there as well, of course. We see even faster substitution rates in pseudogene sequences, for example. So these are lines of evidence that give us confidence that there are different parts of the nuclear genome that are under different selection constraints.
So I'm going to be naughty.
Pardon me?
I'm going to be naughty.
Okay.
He's gonna be naughty.
All right. The gloves come off.
What number could you put on it, and is it something you'd be willing to test in the lab?
For mitochondrial rates?
For any species.
For any species.
So I'm claiming I can make a prediction for cats.
Sure.
Can you?
Or for the cosan, for example. I'm actually very fascinated to see you make that prediction, and I'm interested to see if that's, because that's actually, that takes guts, right, to put it on the line like that.
Are you all daring each other?
The trick is...
Is there going to be a devil dog there somewhere?
The trick is that if you're taking functional constraint into as part of the equation and not just sort of raw mutation frequency, then you actually have to have a good understanding of the selective constraints in those different sections of that genome and also in the environment that they're in. And that's something that we don't have ready access to for a lot of different species. So it'd be tricky from an evolutionary perspective to put a specific prediction on that.
So I'm going to be even naughtier. So if you can't make a prediction for it, should it be considered science?
Well, it's not that a prediction couldn't be made. It just requires a large amount of information that I'm not sure that we have ready access to. So it certainly could be done if enough effort was put to it. Absolutely. And we have seen good predictions made, say, like with the incomplete lineage sorting. We predicted the percentages in advance before we looked at the gorilla genome, before we looked at the orangutan genome. And that was because we had data we had the relevant pieces of the puzzle that we could use to make that prediction. So we certainly could make that prediction for mitochondrial DNA, but it would require resources that we don't have at ready hand. But they could be accumulated if enough effort was put into it.
So if I understand the independent lineage sorting correctly, the prediction was after DNA variation was already obtained for these species?
No, it was prior to sequencing their genomes.
Yes, I guess I'm saying we already had genes, other sequences.
We had a
subsections of the
small
genomes.
subset of it, yes.
Yeah.
And we had estimates of population sizes that were part of that. But we had not looked at a full genome sequence. And we were actually making the prediction off of a very small subset of the DNA. Yeah.
I guess my point is, I thought in my mind, being naughty, it seems a little bit like cheating. Can you predict whether an average guy off the street can you predict how far he's going to hit a baseball? And you let him hit 10 times, you get the average, and you say, yeah. How much of a prediction is that? Prediction is, I predict 450 feet, and I know nothing about you. That, to me, is a prediction, I think. That's what I'm arguing I can do for mitochondrial DNA without getting all these. So would you say, in that light, young Earth creation should be treated as science?
Science doesn't matter where it gets its hypotheses from. That's one of the beautiful things about it. You can have you can have a wide variety of where hypotheses come from. And scientists are actually very pragmatic people. They don't mind what your worldview is. They don't mind what your views on God are. One of the things I experienced, and you probably may have as well, as a PhD and postdoc in secular settings, is that what counts in science is predictive power. What counts in science is whether or not your ideas bear experimental fruit over a long period of time. So nobody really minds where the predictions come from as long as they fit with the data that we have and that they make good predictions going forward.
So would you say the young Earth view should be considered science?
Does science, like I said, it doesn't matter to me where predictions come from. That whole demarcation question of what science versus non-science, philosophers get lots of, that's how they get their paychecks on science versus non-science. So yeah, what matters in science is its ability over a long period of time to make accurate predictions.
Any other questions before we go to the audience? Okay. Well, we do have some questions, and so let me just go back and forth. Dr. Jeanson, let me ask you this first one. A watcher has asked this. It said, would the young Earth position expect genetic evidence regarding the fall? Would there not be an expectation of a massive recreation after the fall? In other words, would not young Earth creationism require a massive genetic rewrite, given what young Earth creationism thinks happened to animals, that they were transformed from plant eaters to carnivorous? If so, if the answer is yes, what research is being done or exists to support this claim? That there would be a massive alteration of bodily morphology and complete system rebalance. Does that question make sense?
And it's a common question. I'll restate it this way. So young Earthers would say no death before the fall, including animal death. No carnivory, put it that way. No pathogenic E. coli, at least no disease, that sort of thing. So where does this come from today? What's the origin of these particular species and structures? Where did lions get the ability to tear flesh? Where does pathogenic E. coli come from? Where do viruses come from? And there's 4 major explanations, none of which, so the answer is basically no, I'd say, massive genetic rewiring. Here's why. You can get pathology simply by changing the environment. E. coli in the gut is good, E. coli in the blood is bad, potentially lethal. So you can find, forgetting the name of the bacteria, there's soil bacteria that do fine, in our body bad. So changing simply the balance of the environment, the harmony of the environment, internal environment, external environment, can lead to pathology. Degeneration. You've got pathogens that are dependent on us as hosts or other creatures as hosts. If they once were free-living, that may not have been a pathology. So you've got change in environment, you've got degeneration as an explanation for pathology. You've got repurposing. I might be wrong in this story, but I want to think the history of the Nobel Prize. Did Nobel invent TNT? I want to think he thought he invented something hoping this would end all wars, and then he saw what his technology was being used for, felt terrible. But so funded the Nobel Peace Prize. Maybe I've got this story totally backwards. Anyway, the point is, you can use TNT for good, you can use it for destructive purposes. You can use sharp teeth to tear flesh. You can use it to open coconuts and watermelons. Not that there are necessarily those plants around back then. It may have been different. The point being, you can get aggressive behavior simply by repurposing it, whether it's good or bad, it depends on how it's used. So that might be a simple explanation. Some might say, well, there's a whole digestive system, short for carnivores, long for herbivores. Pandas have a short digestive system, and they subsist on bamboo. So it's not quite the relationship that we necessarily predict it to be, and we're looking at one point in time. And then last is, maybe more related to the question, latent genetic information. Here's how I'm going to derive it. There are some plants, there's different types of photosynthesis in different types of environments, whole biochemistry, whether you're in the desert or the tropics. And you might think, from a young Earth perspective, God must have created these separate, totally different biochemistry. Well, it turns out some of these plants can interbreed. So you start dialing the clock back and saying, okay. And sometimes the difference between the two is just a simple genetic switch. So going back to this idea of front-loading genetic information. Now this will get into Calvinist theology. So let's step on some toes. There is the potential, depending on your theology, that God could have front-loaded information that could have been switched on after the fall. And that's all I'm going to say about that theological topic.
Can I be naughty for a minute?
Yes, go ahead, be naughty.
I kid a little bit, but We see exquisite adaptation for carnivory in the fossil record in present-day animals. I would have a hard time looking at an 8-year-old boy and saying T-Rex ate coconuts. I really would. I'm just curious what you think of that. I'm sorry, that's a little off the cuff, but yeah.
No, and that's a legitimate question, and you could think of it, here's maybe my first response. You've got lions who seem designed to kill, and we'd say are relatives of house cats. So how much of, and I'd say most of those differences from a genetic perspective were front-loaded, and it's through a process I didn't have time to get into, the result of post-flood speciation. So the first question in my mind is, where in the speciation timeline do they arise? And who are their relatives? Yes, there's things that look designed to kill. And so a design argument would say God made them that way to kill. And that's a strong argument. And that then gets into the theological question. And I won't give away my theology, but I'm comfortable with God knowing what's happening and front-loading it in and seemingly making them designed to kill. So I'm fine with saying no coconuts. Yes, they look designed to kill. And we've lived in a sinful world from a young earth view for most of our history. So we're looking at judgment. And God has exercised judgment repeatedly throughout history. On a global scale, we'd say, from a young earth perspective. So it's a harsh reminder of what we'd say. It goes back to the Adam and Eve question. How's that for a tie-in?
So are you comfortable with the idea that the elephant walking through the Garden of Eden maybe stepped on some insects and killed them?
That gets to a fuzzy issue of where do we draw the line of life versus non-life? What's the nefesh, basically?
And I'm glad to, I'm glad to have that theological discussion with you.
Or should I say it's a squishy line if it's
go.
stepping on?
Squish away. All right, let me. Since we're talking theological questions, Dr. Venema, one that you have gotten a lot, several have asked. You've done a very powerful. You've done a very good job of making a very powerful argument for that change is on a gradient, that it's gradual, and this is true of the hominids. And so when we ask that population of 10,000 that were, what was their previous generation, and they're just imperceptible in the changes. Then where does human uniqueness in the Imago Dei fit into this?
Great questions, absolutely. And biologically, it's a very difficult answer. But biology isn't the whole story, right? So even among Christians who accept evolutionary creation, there's a range of views on these topics. Some hold to some sort of punctate event where God calls our species into relationship with him. And typically they would view the Imago Dei as a relationship, that it would not be predicated solely in our biology, but it would be this relationship that we have. So we're called to be stewards of creation. We're called to subdue and rule, as we see in Genesis. So image in that case is then not so much connected to our biology, although, it's going to have to take a certain amount of biology for us to have a relationship with our Creator and fulfill that relationship and that stewardly responsibility that we have. One of the reasons why I'm a bit uncomfortable in connecting image to particular biological characteristics is that we can look at human variation in the present day, and any characteristic you happen to pick, you can find individuals in the present day who are outside of that or on the edge of that variation. So we can say, well, okay, intellect is the part of the image of God. As a species, yes, we need a certain amount of cognitive capability to relate to our Creator. But then we have individuals who have severe developmental disabilities, and they are as much made in the image of God as anyone else. So I prefer to look at image of God as a relational aspect. And among evolutionary creationists, some are comfortable with that relationship happening gradually over time, or coming into being gradually over time. And other evolutionary creationists feel that there would have been a sort of a punctate event where our species was deliberately and punctately called into relationship with God. So there's sort of that range of diversity there.
Okay.
Yeah, good question though.
Any comment or question on that? Okay. Then back to Dr. Jeanson then. You talked about it's not a cloning event. that Adam and Eve were not a cloning event. However, Eve was taken from the rib of Adam. So, one observer asked, couldn't that or shouldn't that be understood as a cloning event?
How much time do we have left?
Just a few minutes.
Because there's the obvious question that no one's asked yet, but I don't want to miss it. I'll answer that one quick.
Okay, well then, okay, go ahead.
Yes, she's taken from his side, so that would imply that if Adam was created with two different versions of his DNA, she would have been virtually identical to his two different versions, except for sex chromosomes. Obviously, she's female. And what's interesting is, if you look around the globe, most of our DNA varieties come in one of two versions, fitting that sort of idea. So, is she a clone of Adam? Yes and no. There's all sorts of differences within Adam, differences within her. So, you could call it that, but usually when we talk about cloning, we're saying making a genetically identical version. Is she genetically identical? Well, if you want to call a whole bunch of differences identical, I guess so. Yeah, so there's that element, but it seems to fit what we see in biology.
All right. Any question or comment?
So I'm just curious on that point. So you're saying that there would be new created differences in the process of making Eve? I'm just trying to understand it from a...
No, in that sense, it might seem to be like cloning. She's made like Adam. She has, except for the XY, obviously, because she's female.
Adam has an X, so all you need is two copies of that X, right?
Well, I guess I'm saying not completely identical because she's got to have something that makes her female.
Sure.
But otherwise, would his chromosome 22, his two different pairs, look the same as Eve's? Potentially.
Okay.
But both of, Adam's number one of that pair and number two of that pair look different from each other, as do Eve's from one another. And then that'll lead to all sorts of, the potential for variety is incredible.
Okay. Interesting. I'm just curious. I'm curious about that just from a technical point of view, because some young Earth creationist materials seem to posit 4 different starting points, but what seems like you're saying is that there's just really 2. So I'm curious. I'm kind of just...
A priori, you might expect that. Rob Carter's one who pointed out to me saying, you do have that from the rib. Does that mean it has to be the same? Maybe, maybe not. And then he said, well, most of our DNA differences come in 2 versions, implying then it goes back to just 2 different versions rather than 4.
Interesting.
We're running out of time, so let me ask you the question that you started with, that you asked to Dr. Jeanson. What would cause you to do a rethink? What type of evidence or what kind of new development might make you say, okay, in other words, what kind of discovery or hypothesis might Dr. Jeanson present that you'd say, I'm going to have to really think about this?
Interesting. Well, as a scientist, scientists are always open to new information. It's actually interesting, Nathaniel, your answer was very, very long and detailed. Your colleague's answer a few years ago was not. It was very brief, and the answer was no evidence, right, because it's a scriptural issue. What I see on this issue, so yeah, scientifically, if young Earth creationism, over the next decade wins the day scientifically, absolutely, scientists go with the evidence, no problem. I don't see that happening. There's lots of lines of evidence that we haven't talked about tonight, geological evidence, those kinds of things that I think make the young Earth creationist position scientifically untenable. But say, it's conceivable that, scientists are always open to new evidence, so it's conceivable scientifically that it could shift, I suppose. But I would be extremely surprised. I would also be surprised if we, could I conceivably shift my views on heliocentrism? You bet. It could well be that there's a better model out there. Now, that better model is going to have to incorporate the data that we already know about, and it's going to have to do a better job of explaining it than the mainstream data, or the mainstream models of explaining it now. But any scientist would always put a caveat on anything. Even things that are extremely well established in science can be shifted, but I don't think we're going to change heliocentrism anytime soon, and I don't think we're going to change evolutionary biology anytime soon either.
Well, these men have been speaking to us for 5 hours. You've been a great audience. I want to commend you for how you've stayed with us this afternoon and evening. Also, these men, I think, have done a very good job. It's been very helpful for us to understand their respective positions in a way that I don't think we did before. So let's show our appreciation to both of these men.
Thank you. Thank you. Appreciate it.
God bless you all, and we're dismissed. Be careful going home.
Thank you.